A very good afternoon to all of you. The organizers again welcome you to the post-lunch session of 3 days Pulse Webcon 2021. This session will be on post-abiotic stress and its management. Just before this session, we had the celebrations of World Pulses Day and the opening session of Pulse Webcon 2021. In the gracious presence of Honorable Union Minister of Agriculture and Farmers Welfare, Shri Narendra Singh Tomarji. Now for this session, I have pleasure in inviting Dr. P.S. Basu, Principal Scientist and Head, Basic Sciences Division, ICR IIPR Kanpur, to kindly handle this session from Kanpur and initiate the proceedings. Dr. P.S. Basu, please.
Good afternoon, everyone. I'm just going to initiate the poster. This is the sessions on abiotic stress management. As you know, that is a very, very important aspects of the pulses, because pulses predominantly grown under the rain-fed condition, where water is the most valuable resources. Now you see that if we're talking about the drought, it is an integrated part of the pulses crop. If you want to enhance the pulses productivity and drought problem or the constraints we have to address. Apart from this, we have the major stresses on the heat because the climate change, and during the the terminal stresses, and most of the crops and the pulses are exposed to temperature, it is just exceeding the below threshold level. So heat is another major problem. The first and foremost is drought, followed by the heat. So these two aspects will be addressed. Our distinguished speakers are willing to address, and I think that these two aspects are well covered. Apart from these things, we have the distinguished panelists. I briefly introduce the panelists. Our most respected, the moderator, Professor Uday Kumar, and he is Professor, Department of Crop Physiology, US GKVK Bangalore, till date. He is the Coordinator, Biotechnology Department, US Bangalore, member PSC, Plant Sciences, and DST. He has many awards and honors in his credit, including the R.D. Asana Medal, ICR Best Teacher Award, and also the Fellow, FNAS, National Agriculture Academy of Sciences, then FNA, Indian National Science Academy, Delhi, FASC, Academy of Sciences, Bengaluru, FNSC, National Academy of Sciences, Allahabad. He has International Society for Plant Molecular Biology, Indian Society of Plant Physiology. His research areas are crop physiology, photosynthesis, crop productivity, stress responses of plants, physiology of water use efficiency. Now coming to our distinguished panelist, Dr. Venkat Sharulu, as you know, that is the former Director CREDA and Vice Chancellor Basant Rao Nayak, Marathwada Krishi Vidyapith, Parvani. He has many prestigious awards and honors in his credit, notably Basant Rao Nayak Award of ICR, Fakhruddin Ali Ahmed Award, Hari Om Trust Award, International Crop Science Congress Award, Adjunct Professor Nam Hyderabad, as well as he had been chairman QRT RAC for the ICR of the various research institutes. He is member, Scientific and Technical Appraisal and Advisory Group, DBT, member, Genetic Engineering Appraisal Committee, member expert committee, and he is the main instrumental to launch the NICRA project, which operated more than a decade or so, and lot of the works related to abiotic stress has been performed, has been completed under this NICRA project. He is fellow Indian Society of Oilseed Research, Arid Zone Research Association of India, Indian Society of Dryland Agriculture, Andhra Pradesh Academy of Sciences, member New York Academy of Sciences, is a life member Association of Microbiologists of India, life member Indian Society of Agrometeorology. His research areas climate change, abiotic stress management, and soil microbiology. Another panelist is Dr. Chinnaswamy Vishwanathan. He is head plant physiology, IRI, New Delhi. He is postdoctoral experience in Tucson, USA, University of California, USA, and visiting scholar at Harvard University, Boston. He has been honored with many awards, including the J.J. Chinoy Gold Medal, Indian Society of Plant Physiology, G.S. Sirohi Award, Jawaharlal Nehru Award, and had Boy Scout Fellowship, DST. He got the fellowship from the National Academy of Agricultural Sciences, Fellow of Indian Society for Plant Physiology, and Associate of National Academy of Agricultural Sciences. His specialization particularly is abiotic stress physiology, stress signaling, microRNAs, phenomics, transgenics, and genome editing. He has 20 years of research and teaching experience in plant physiology and published about 150 peer-reviewed publications with a total citation of more than 14,000. He established state-of-the-art automated plant phenomics facility at IRI, introduced a course on advances in plant phenomics for PG students, and identified genotypes and QTL for water use efficiency and nutrient use efficiency in rice and wheat using phenomics. Another panelist is Dr. Jagdish Rane. He is presently head the drought school in ICR, National Institute of Abiotic Stress Management. He visited International Center for Tropical Agriculture, SIAT, Colombia, and became the leader of the plant phenotyping team for evaluation of gene technology for drought tolerance in rice. He started employing image-based tools for assessing plant responses to drought under phenotyping platform. He employed root trait-specific molecular approach to impart drought tolerance in rice, mega variety IR64, which is published in Nature Genetics. He established high-throughput plant response characterization facility called Plant Phenomics at Niyasam, Baramati, and is main instrumental in optimizing protocols for assessing drought responses of mung bean, chickpea, pigeon pea, soybean, maize, sunflower, etc. under ICR National Innovation on Climate Resilient Agriculture. He presented his research accomplishment in USA, Mexico, Brazil, Australia, Japan, Philippines, Egypt, Germany, and France. Recently, he has also launched project for exploring underutilized crop for the drought-prone areas. He was awarded gold medal for academic excellency as PG students and International Centers for Tropical Agriculture, Colombia. He has more than 80 research publications in national and international journals and an edited book on abiotic stress management. With this brief introductions of the panelists, our distinguished panelists, I will request our moderator, Professor Uday Kumar to take over the session. Professor Uday Kumar, please.
Good morning, all of you, for today's afternoon's interactive meeting on abiotic stresses and the management. And I will take this opportunity to also welcome Dr. Basu as the panelist for today's afternoon's meeting. He's a very modest person. He has not given us the requisite input that is needed to tell about him, but he is one of the leading scientists in the area of the pulses. More often, he became a synonym to pulse physiology in India. His contributions are excellent, and more than anything else, he has been the avant-garde and taking the flagship to bring in requisite amount of the interaction that is needed amongst the scientists working in the pulse area, mainly because of the fact that pulses, like other core cereals, are the rain-fed crops. Pulses are the ones which are grown under stress environment in terms of drought, moisture stress. And in addition, in recent years, because of the global climate change, we have issues in relation to the high temperature, especially for the SISA coming into the picture. And there is today's topic is very apt that to look into the insight that likely to be provided by today's, what we call it as the invited talks from 3 distinguished scientists working in this area over the last several years. And in addition to that, I would like to specifically welcome the other panelists and very distinguished scientist and administrator, Dr. Venkateshwarlu, who has been heading the CRIDA and also responsible for the NICRA program, which is really giving us remarkable outcome for the benefit of the Indian farming system. And in addition to that, 2 of my friends, Dr. Chinnaswamy, who has been doing excellently, Vishwanathan, and contributing extremely in several areas of plant physiology, more specifically the phenomics, and in addition to the molecular plant physiology aspect. And finally, we have Rane, a good friend of mine, who is an excellent person, an interactive personality, taking actually the phenomics and interacting with the genomics aspects, and made a significant contributions in the NISAM, in the Institute for Abiotic Stress Management in Baramati. With these few words, I would like to know from Dr. Basu that we have left with approximately 50 minutes from now. And we have 3 speakers. I would request actually the speakers to complete their presentation in about 12 to 13 minutes, so that we have, at the end of the presentations, we'll have enough time to interact and to know more about the specific aspect that they are proposing in their presentations. The first speaker that we have for us is Dr. M.S. Seshashayi. I'm not seeing easy arrived already. Dr. Basu.
Yes, sir, I'm here, sir.
Oh, Dr. Sesha is here. I don't have to really,
hello,
it is difficult for me to introduce Dr. Seshai. First of all, I would say formally I introduced him. He is the professor of crop physiology in the Department of Crop Physiology, University of Agricultural Sciences, Bangalore, right? And of course, Seshai is known to be a person who initiated and established a national facility on stable isotopes, for which he has an opportunity as a Boy Scout fellow to visit the stalwart in this area, Dr. Gram Farquhar in Australia, with whom our department has been collaborating for the last 4 decades. Having gone there, Dr. Sesha is also a member actively participate with IAEA programs on utilizing the stable isotopes. But more than anything else, being a person in the area of the key person in our drought center of the Department of Crop Physiology, he made significant contributions in the area of phenotyping for the different drought adaptive traits. And more recently, he has gone in terms of identifying, adopting this phenotyping background that he has gone into the genomics and then developed insight in identifying the QTLs for the important drought adaptive traits. As an outcome of it, he recently released a variety based on the physiological breeding called Daksha in rice, which is occupying quite a few areas. His contributions are quite a few. And I will not go into the details except for the fact that he is recently elected as a fellow of the Nasi Academy of Allahabad, and we wish him many more such laurels to become a fellow of the other important science academies of India. I will not come in the way in a big way, and I will straight away go. And he has been contributing in addition to the scientific contributions, and also participating in different review committees in DBT, DST, ICR, and etc. We look forward, and he's a good teacher. He is the best teacher award from the INSA. Now, may I request Dr. Shesha to go ahead.
Hello?
And, yes, please.
Professor Uday Kumar, can you hear me?
Yes, I can hear you. I can hear you.
So the program, just our session will continue up to 4:30. That is a 2 hours period.
Oh.
And then in between there is a addressed by the Ali Abu Sawa, the Director General of ICADA. So that is a 15-minute duration that will be interrupted. And it will again continue after his address. So, sir, okay. It is not a 50 minutes. It will run up to 4:30.
In such case, what is the time allotted to these speakers, Dr. Basu?
Half an hour, sir, lead lecture.
Half an hour. Okay. And half an hour for the lead lecture, that is from Dr. Seshasai, right? And let us come back with regards to the others later. Now, without losing time, now may I request Dr. Seshasai to give his presentation on improving drought adaptation in pulses. Dr. Shesha, please. Share your slides and then initiate your presentation.
Yeah, I already shared the slide. I think you can see it. Dr. Basu, can you see the slides?
Yeah. Nirja, Soumya.
Thank you very much, sir. I should, before I start, express my sincere gratitude to Dr. Basu and IIPR and an excellent conference that has been going on since yesterday. I had the opportunity to listen to some of the fantastic speakers, lovely information that is coming out of the kind of research that is going on now. And the other distinction is today, being the World Pulse Day, I think it's an important thing to sort of talk about this species. And I also thank Professor Uday Kumar, and it is a blessing for me to be introduced by my guru. So, okay, so without wasting my time, I thought it was a test match converted to IPL. So to hurry up, but then I think we can go slowly in this, but then I'll not take more than about 20 minutes, and then we'll have sufficient time to discuss at the end. Like, before we go into these slides, there is one thing that I wish to say, that, my experiences with working actually in the pulses has been not very, very high, in the sense, like we have been concentrating mostly on rice, but then we did do a lot of work with Rajiv Vashne at ICRISAT on chickpea and pigeon on groundnuts. But then going ahead, there is something that we would like to now talk about, is that there is some kind of a shameful statistics that we give, that every second child in India is a malnourished child. And every third child out of the 5 children in the world is some kind of a malnourished child is from India. So now the problem appears is that when we look at this kind of things, and even in if you look at the reasons for this, the major reasons for the stunting and the wasting that they talk about in the medical terms comes mainly because of protein malnutrition. And in India, about 38 to 40% of the children that we have are under size in terms of their height. And when you look at this question as to why it is like this, there are two major things that emerges: the protein and the mineral malnutrition. That will be the primary cause for these kind of abnormality that we have. I mean, there is a kind of an importance now that when people in India had a problem of dying for hunger, there was a lot of emphasis on cereal research. But then now, if we want to really be healthy, it has to be sort of shifting towards the protein sources. Now, there are two questions that we have in the moment when it comes to the proteins. One is the availability of the quality foods, and then the most important thing seems to be the affordability of these foods in Indian populations. And it's mainly because of the poor income that we have and poor education status that we have in the Indian, generally in Indian populations. And the poor quality and the amount of protein that we have in the food and their digestibility. We've been working with the medical sciences and do clinical nutrition to now understand whether the vegetable-based or the vegetarian-based diets are digested at all and the proteins are available for human beings. Now, the question that we have, therefore, is can these pulses reduce this burden on the human population in terms of health? And when we look through the literature and then understand the kind of work that has been going on in pulses, the answer to this question, whether the pulses can reduce this burden, is yes and no as well. It cannot do the job, and let's see, both of them, we will try to now analyze it and then come back for a discussion. Yes, I say that because when you now compare between any of these cereals, the pulses have several-fold higher protein content in them. And that's a very, very good kind of things. But then there is some issue that is associated with the digestibility of the proteins from the grams or from the leguminous sources. It's about 56 to 58% is digested. But then if you look at the rice and wheat, it's about 70% of the food is digested. Proteins are digested by human beings. But then for sure, the protein content of the grains that we eat from pulses is much higher than that of the cereals, and that's the reason why we should be really worried about it. I say no to this question that the pulses cannot do the job of really overcoming the malnutrition, especially protein energy malnutrition in India, is mainly because of the fact that the productivity has remained very, very low in India for a very long time. And there are some interesting statistics that I got a few days back to share with you. I'm sure I'm talking to a very qualified audience, such as the Indian Institute of Pulse Research and all the pulse scientists. I may be wrong, correct me if I'm wrong. This was taken from the Ministry of Agriculture, Government of India. Now, what they say is that look at this, India is one of the highest producers of pulses in the world. But then yet we are protein malnourished is something very, very strange. The problem is it's because of the area that is actually diminishing over time, and no other country has so much of area. But then the question is that when you look at the yield per hectare, India is among the lowest, and the only other country which has a lower yield than India is Niger. I think these African countries are very impoverished, and India, I do not know why the yields have remained for a very long time, only around 500 kgs per hectare, or 500 kgs per acre it could be. Now, there are two questions that I always ask to bother. One is the pulses, which are grown in mostly the water limiting or very highly impoverished conditions, has the two major problems. One is the lower yield potentials. That is, even when you give them the most optimal conditions for growth, the yield potentials are low. And these pulses are unfortunately, or perhaps fortunately for us to do some experiment, there is a lot of opportunity to work, is that they are very sensitive to both biotic and abiotic stresses. And the two very, very important stresses that we need to talk about among the abiotic stress are heat and drought. Now, if you look at the source of pulses the pulses as a source of protein, and then when you look at the amount of water that is required for this, though I said in the beginning the rice protein is more easily digested by human beings than the pulse proteins. But if you look at the water requirement to make that, it is called the water footprint that the protein would leave. You have the pulses having something close to around 7,500 liters per kilogram of protein. And when you look at the 6% protein that we have in rice, though it is little better digested, then the amount of water that it requires to produce that 1 kilogram of protein, I'm not talking about 1 kilogram of grain yield in rice, but then it requires about 60,000 liters of water. So that means for getting the kind of protein in India, about 60% to 68% of the protein requirement comes from cereals, which is at a very huge cost for water. Therefore, I believe pulses and the protein that we get from the pulses are the most affordable kind of sources which can be sort of exploited and then improved. But before we harp into that kind of a question of whether how is it that we can do this, there are a few questions must be answered. Can we really decrease the footprint at all of these sources in terms of the protein? Decreasing the water footprint seems to be an important objective globally, and especially in countries like India, where water is a crisis. And when we depend on the monsoon and the monsoon becomes erratic because of the global climate changes, and then we have real problems in terms of achieving both food security as well as a nutrition security in India. Now, having that is one part of it. Other bigger part that comes is, can we really increase the yield potentials? Now, yield potentials, I talk about the potential that we have for the yield when there is absolutely no limitations for any resources. And that conditions can we still improve it from whatever the average that we have is from 5,000 to a little more than that. Let's say if we can reach something like 2 to 3 tons per hectare in pulse productivity. Can we improve the stress tolerance? That seems to be one of the most important questions that's being addressed in this session as well. And then we will have an opportunity after sharing my thought about this. We can also listen to the other speakers and then have a panel discussion with the distinguished scientists that we have. And of course, this is one thing that we always talk about these days is, can we really increase the digestibility of the pulse proteins at all in human beings? Now, one of the mechanisms that has been studied very recently, in 2018, this paper came in one of the MDPI journals. Now, what they do is to reduce the water footprint of any grain. They have studied a number of species or crop species here, including cereals and oil seeds like canola and sunflower, lentil and chickpeas pulses. Now, what you can see here is there is a reduction in the water footprint, whether it is in terms of the water footprint for yield or water footprint for protein. There is a reduction in the water footprint if the yield increases. This is where the catch is. Are we decreasing the water footprint by decreasing the water requirement of the crop, or are we decreasing the water footprint by increasing the productivity that we have per unit hectare for whatever that the resources that are given? From this data, it looks very amply clear that the reduction in the water footprint predominantly is controlled by the increased grain yield or increase in the protein yield. And increasing the protein yield in the pulses may be very difficult. Like, as you can see, that the lentil and chickpeas are somewhere here. Whereas by in other kind of thing, other kind of crops like cereals and all that, there is a possibility of increasing the proteins. But then the only possibility, therefore, seems to be in the case of pulses is to increase the productivity per unit area so that we can decrease the water footprint and the water requirement of the crop under this. As I said before, despite the kind of effort that is being done in many of the institutes and in the agricultural universities and so on for improving the yield levels and understanding the mechanisms of stress tolerance, both biotic and abiotic stresses in pulses, the increase in yield has unfortunately remained very, very stable in the last 25 years. But what has happened is from the 1950s to 2000, there's been a double or triple in the case of rice. And this is something which is understandable and also bothering. Understandable because there was a lot of effort in rice on improve the rice and wheat productivity, mainly because at that time when there was famine and there was a need for us not to die of hunger. But then are we now happy with the kind of scenario that we have for the yield level that we have for pulses like this? Are we ready to die of malnutrition than of hunger? That is the same question that we have now. But if you look at this graph, this was taken from the Ministry of Agriculture once again, and I computed this data now. In the 1950s, pulses used to form about 85% of the total grain that were produced in India. But then it has slowly decreased, not because of the reduction in the production of the pulses, but it is mainly because of the significant increase in the production of other crop species. The proportion of the pulses that we have in the Indian food grain basket has come down to something like 30-35%. Now, this is something which is a question to bother. If you understand the plant physiology of it, there is something why in the cereals it can happen and why in pulses the increase in productivity is not happening is a question. But if you, again, the same thing to talk about the yield potential and the realized yields as what we are talking about in terms of pulses that we have, which is 500 in the farmer's field as a national average. But then we can look at this, rice is about 2,500 to 3,500 tons per hectare. But it is possible that we can get very high yields. I think Rajiv yesterday must have been telling you that there are possibilities of increasing the yield potentials even under irrigated conditions, that there is a possibility of increasing the productivity of chickpea. And then there's always this possibility. This is all some data that was published by ICRISAT some years ago. And what is it that we can say is the reason why the yield potentials have not increased to the levels of the cereals, and why is the realized yield in the farmers' fields so low? One of the reasons that we have, and we can answer this in two different ways. Now, if you look at the very simple physiological mechanism that we talk about in terms of how to increase the productivity, we look at whether the crops are growing well, we look at the crop growth rates, as well as we look at how much of the biomass that is produced by increased crop growth rates are actually taken for the harvestable part, that is what we call the harvest index. Now, if you look at this here, the increase in the harvest index has not been very significant in pulses, and the relationship that we have with the yield is not also very. I mean, it has to be strong, but then somehow there has been not a very, very significant increase in the harvest index over years. On the other hand, if I increase the assimilation of carbon dioxide or assimilation of carbon by photosynthetic mechanism, which will lead to increase in the crop growth rates, there is an increase significant. We're talking about 400 ppm, which we have now, but then the CO2 concentration that we have for the crops is slightly lower in terms of the internal CO2 concentrations. But if you see this, there's a very steep increase in the possibility of increasing the assimilation rate if the CO2 concentration increases. Here that we have is yield. As the CO2 concentration increases, there is a significant possibility of increasing the productivity. This is not a very recent, about four years back, this kind of experiment was done, and despite the fact that there is a possibility of acclimation to long-term exposures of the plant to elevated CO2, there is still a significant increase in the productivity, suggesting that it could be the source, which is a big limitation. And the problem with the harvest index not really being increasing is another thing that is associated with the sink. So we have two very important mechanisms that we need to address. One is the source, which is making the carbon assimilates, and then to the transport of them or translocation of them to the sinks, which are to be now increased. Now, what we have here is to show this: if the CO2 assimilation is increasing at higher light intensities, there is a much larger increase in the assimilation of carbon dioxide at any given CO2, which is actually much better in pulses than in cereals. Cereals, you see that this particular gap between the low light and the higher light intensity for CO2 responses is not that very high. And this suggests, once again, that the pulses suffer solar energy utilization efficiencies. Now, we have compared these things in some of the experiment that was done by Professor Uday Kumar's team quite some time back. What they did was to now compare the energy utilization efficiencies of several crops. Like rice, for example, is very high when compared to the cowpeas and pigeon peas and soybean, or somewhere at the end here on the right side, suggesting once again that the decrease in the crop growth rates is perhaps mainly because of the fact that the crop is not able to now utilize the right energy as efficiently as the cereals would do. Is it the canopy architecture? It is the chloroplast mechanism? Is it other kind of things? We will have to see this. So if the potential yield is related to the crop growth rates, and the crop growth rates are governed by the radiation use efficiency, and of course multiply that with the partitioning of the biomass into the harvested parts, is the way in which the physiologists would look at. Now, if you look at the radiation use efficiency, there is some amount of light that is intercepted, and there's a huge proportion of the light energy that is used efficiently by the system, and that leads to the carbon assimilation by increasing the efficiency of carboxylation. So we are talking about two efficiencies here. One is the energy utilization efficiency in terms of the radiation, and the conversion of the inorganic to the organic carbon by the carboxylation efficiencies. Now, when you look at this here, if I increase the solar radiation incident on the crop, there is a reduction in the efficiency with which the radiation can be utilized. And this is something which is interesting. And this decreases very sharply in rice, but you can see in pulses that it doesn't decrease as much as that we have, which means there is an opportunity for us to sort of look into whether we can increase the energy utilization efficiency of our pulses so that we can increase the yield potentials to increasing the crop growth rates, is what I said. The other kind of thing that we talk about is the interception of the radiation, and unlike the cereals, which have some kind of an erect canopy architecture, you have a sort of a shading kind of a canopy which is drooping here in pulses, and that prevents the radiation to get inside this. What people have done is to now understand whether the photon flux density that we have inside the canopy, that is, you have the first, second, third leaf, and if you compare with this kind of a situation that we have here, CO2 assimilation in terms of this first leaf is high anyway. They both are getting the light completely. But in the case of this scenario, you have very low radiation that is going to the bottom. And therefore, the total amount of radiation that is intercepted by a physiologically, functionally capable leaf is going to be less in the case of pulses. So is it possible that we can look at some mechanisms by which we can now change a little bit of change the architecture a little bit so that we allow more radiations to come in, or make the lower canopy leaves to be very responsive to flecks of light that can come when leaves move and sway when there is wind or something, and also to be efficient in terms of utilizing low energy radiation. So we have, again, a lot of opportunities to look at. If you looked at the CO2 light response curves, with increase in the PP, the photon flux densities, whether the photosynthetic rates can be increased. There is this slope, initial slope of the light response curves, which is considered as the quantum efficiency of the leaf for photosynthesis. Significant differences exist among the germplasm. So we need to sort of look into these kind of things and then understand how to exploit these kind of things for increasing the productivity of our plants. The other kind of thing that we have is a very significant relationship that the pulses have and compared to the cereals is to diagonally adapt to the kind of changing environmental conditions, weather conditions within a particular day, which is predominantly driven by the vapor pressure deficits. Now, this particular work which was done in Moonbeam suggests that there is a very significant reduction in the stomatal conductance with time in the day. You can see that by about 10 o'clock, these guys are started coming down already. And then the photosynthetic rate, though it stays there a little bit higher, but because of the low light that we have, the photosynthetic rates of pulses on an average in a particular day is generally very, very less. Therefore, it is again important that we understand these mechanisms which will actually lead to improving the carboxylation efficiency and also the radiation or the light use efficiencies, so that we can improve the productivity of these things, of the pulse crops. The other thing that happens is the senescence of these leaves, which in many crops like maize and sorghum, people have worked into understanding what are the genomic resources that we have in terms of the QTLs and genes that are associated with the maintenance of the chlorophylls in the lower canopy leaves, which are shaded basically, and the shade-induced senescence can cause a lot of problems. And you can see that as the leaf age increases, there is a significant reduction in a number of physiological parameters which decrease the photosynthetic rate. It could be the rubisco activity and the chlorophyll content, the total photosynthetic activity. I mean, to cut the long story short, as the leaf age increases, in any species there is going to be a decrease, but in pulses this particular decrease is much steeper. More significant in pulses is what we say is mainly because of the fact that the lower leaves are anyway shaded, and with their age they are going to be more senescing, and maybe their adaptation to low light intensities has not been really investigated and thoroughly understood. So we look at one of the other mechanisms to now improve that would be to now find out if the CO2 fixation can be improved by understanding such physiological factors as the stomatal conductance, so that the CO2 can come inside. By looking at the stomatal conductance, we must also remember that the stomatal conductance, when the stomata are open, it can also promote a lot of transpiration, and so we will have certain problems for water limiting, rain-fed conditions of this crop. But improving the mesophyll efficiency in terms of carboxylation, diffusion of the CO2 through the mesophyll cells, and then we need to look at, these are all very clearly understood basic science mechanisms, but I think it is important with all the kind of development that we have in science. These are all very much possible these days to now do this. And then I'll show you some of those mechanisms which Professor Uday Kumar was also mentioning, that the phenomics platform that we have at IARI and the phenomics platform that we have established at US Bangalore, which I will try to show you some slides at the end, will perhaps become a very, very important strategy by which we can now screen a large number of germplasm for variations in all these traits. One of the things that we have during this kind of dry conditions, which I also said a little while ago, was that the pulses are more sensitive to the vapor pressure deficits that they experience during diurnally or in seasonal variations, and especially under rain-fed conditions. And you can see that the VPD sensitivity as the temperature increases, which is definitely going to happen in the years to come. Pulses are more sensitive to this than the other cereals like wheat and rice. So this is another challenge that we have, and I mean, it is sometimes disheartening to say there's so many bad things about the pulses, but then the moment we know what is the problem, and then I think we can really rectify those things and go ahead in terms of improving the productivity. And there are a lot of research that has been done to show overexpression of some of those photosynthesis genes, especially the RUBP carboxylase activase, and to keep the activation state of this enzyme high, there's been a significant improvement in productivity. I'm not saying anything that we should adopt this one, at least for the time being, because I mean, we don't have a mechanism to now allow these genetically modified plants for consumption by human beings yet in India. I hope it changes. That's my personal opinion, so let's not be quoted on that. But when there are a lot of such examples I can say. So to go ahead with this, it's not necessary to bother about in the sense to emphatically suggesting that increasing the photosynthesis has a significant influence on net carbon assimilated by the total canopy, and therefore improve the crop growth rate to increase the productivity in pulses. So there are a lot of such information that are available in the literature. But there is one more problem, that's about the source. So you have the concept that there is a lot of problems in the source, which is making the carbon assimilates available for reproductive growth. But then the problem is there's a significant flower drop, and the pod filling is not very complete in pulses, which is another challenge. And we can see this when compared to, I mean, cereals do have some of these kind of things which we call a chaff, but then there's no flower drop as such. But we can look at that there is so much of about 30 to 70%, maybe sometimes as high as 90% of the flowers drop, maybe because of many factors that are associated with it. A lot of science has gone in again into this. There's a lot of basic science that people have worked on this. So what we have is that we have certain specific tissue that we have in the dicots, especially these pulses, called the abscission zone. And the abscission zone will go through a lot of these developmental changes which are hormonally regulated, and they respond to the kind of changes that influence, such as the VPDs, temperatures, and relative humidity of the air, influence the plant's susceptibility to become more amenable for abscission, and that actually causes them to break off from the plant, and the flower drop occurs. So much of science has been done, and this science therefore can be adapted for improving and to find out if there are any QTLs and the genes that can be discovered for some of the traits or some of the important traits that are both associated with source as well as the sink of the pulses that can be definitely achieved. And very recently things have started. Indians have been the pioneers in sequencing the pigeon pea genome, both at NIPB in New Delhi, and then NICISAT by Rajiv's group and NK Singh's group. And I'm very happy that these kind of things are taking forward in a big way to look at the chickpea, pigeon peas, and all the other kind of pulses in terms of QTL mapping, which I'm sure Dr. Basu also is all very actively involved. But most important thing that comes to is when we look at the stress and the drought conditions that we are talking about, one of the major things that happens is that the tissue turgor, which becomes poorer when the plants experience stress. There are some of the traits that we can really say is to the one that are associated with water use or uptake of water, and then you have some of those factors that are associated with the conservation of water, both in terms of stomata as well as the cuticular axis in terms of preventing the water loss, and the assimilation that are associated. All these metabolisms are in a way very strongly associated with the leaf turgor or tissue turgor. Now you have two options that we can look at as a strategy for improving the pulse crops. One is, of course, with the kind of expansive expertise that we have in the country and in terms of the resources in genome sequencing, and the genetic resources that are available in terms of germplasm, which has been resequenced already. There is a possibility now for going ahead with the genome-wide association studies, which requires one of the very important things. This, as I said, is easy. And with the other thing which is important is the phenotyping. And that is where I think the physiologists should step in and then really make an impact in terms of competing with the genome sciences, and then talk about the phenome and the phenotyping and the phenomics kind of things, which is happening in India now very, very strongly. Look for all the traits that are associated with the yield potential, like we discussed about the radiation and the carboxylation, fruit drop and the flower drop and all that. And then you look at the drought adaptation in terms of what to use and everything. And of course, there is one thing that we need not bother about it in terms of the productivity point of view is the anti-nutrition and the digestibility issues, which are all important. So for these kind of things, QTLs are being identified, and the contrasts are being selected, and then crossing programs have started becoming. But the problem with drought is how are we going to choose a trait that we need? And most important question after that, there's a tremendous amount of information that is available in the literature that we can now suggest. There are a number of those. Yes, sir, I need about 3 more minutes.
Yes, Dr. Shesha, you have to conclude. Yeah?
3 minutes only, sir.
Yes, yes.
Okay. So then suppose we have the choice of this, and then there is so much physiology that is associated with the yield, and what is required is for at this time being is the understanding the stress responses, and to capture the genetic variability that we have in the drought adaptive traits. Very precise imposition of drought stress is essential. That's where the conceptual platform that we established at UAS Bangalore, and I cordially welcome everyone when the COVID situation eases and then we are permitted to travel. Please do come and visit us. And I want you to, I said 3 minutes is because I thought I would show you the small video which will run for about 2 minutes. So these are the high-end platforms where precision imaging, capturing and sensor, the plants go to the sensor on a conveyor belt. And then we also have some of these mechanisms where the sensors go to the plant in terms of mobile kind of gantries. But then most of these things are very highly costly, and then they are inhibited basically. So we try to now develop something which is cheaper at the Department of Crop Physiology, thanks to ICAR for providing the funds for this. And then we developed this mini lysimeter based mechanisms where the containers are grown on a load cell, and then there is a rainwater shelter that protects it. And the irrigation is automatically provided to that. These plants are there, and every individual container can be monitored for water use, and the growth and all those kind of things. For this is automated irrigation that I said, precisely dispensing the amount of water that is required pot by pot. We have an automated calibration system. We don't have to really do this. Everything is semi-automated here. We only have to go there and command it. And then we have a Wi-Fi mechanism to transmit the data directly into the server. And the imaging can also be done in terms of the plant eye, which can go there and collect all the data. And we have the dashboard, and this is some of those data that we have collected recently on all the experiments that we did. And the moment you talk about this, they are grown under the natural conditions. Therefore, we have all the, that's where my boss is. And it is actually suitable for all sorts of crops, and especially pulses. And like Sudhir Kumar said, that we developed this Daksha variety, and then all the phenotyping was done under this platform to now validate this. So this is doing well. I think this particular facilities, such as the phenomics platforms, are the most important need of the hour. So the major features that we have is that the rainwater shelters, like we saw in the video, protects the plants from rain episodes, and in the night time, therefore most of the time these plants are outside. And then the plants grown in the mini lysimeters, which are placed on the load cell balances individually, will have a record of the weight in real time. Every second you can have. And the best part is that we can give a very specific stress regime. you can say that the soil dries up in 10 days or 15 days, or any of those kind of things to mimic a multi-location kind of an experimentation can be done very effectively here, which we call the dry down protocol to maintain a specific level of stress. This facility is highly cost effective and operates under natural conditions is one of the major features of this. In summary, we have made excellent progress in understanding the physiological basis of drought adaptation in many crops, including pulses. And phenotyping has advanced significantly now, and excellent techniques are available. High throughput mechanisms are available. I didn't tell anything much about the stable isotopes and other kind of thing that can be done here, because I wanted to focus on what needs to be done, which can be debated in the panel discussions. And the genetic resources such as germplasm, mapping population, everything is available with the breeders and other scientists in India now. And the genomics-based approaches are very much possible now because of the reduction in the cost of genome analysis and availability of the data that is there, and the computational biologists are plenty now. I think we are in a very perfect situation now to put our hands and efforts into improving the pulse productivity. I should thank the funding agencies that have supported us in terms of developing all the infrastructure like the DBT, SCRB, DST, FIST, and then the Karnataka state government for the RKVY project. And of course, in one of the recent experiment under the Indo-Swiss collaboration in biotechnology, we had a lot of experience in working with the best of bioinformaticians and then genome biologists. Thank you very much for this opportunity.
Thank you very much, Shesha. Though it took a little more time than what's allotted to you, but you have given a very comprehensive picture about the status of the pulses in terms of the need, approaches, and the diverse ways that one could look for to enhancing the potential yields of these crops, both under control and the moisture stress conditions aspect. Dr. Basu, I would like to know that we'll come back to the discussion on at the end of the presentation of all others, right? And during that particular period, and I hope Dr. Shesha, you had to be around, and then try to answer the questions later. If there are going to be any very pertinent points that are going to be made by the panelists as well as the others in this direction. So once again, I'll thank you for an excellent presentation and a very comprehensive presentation and a thought-provoking presentation, Dr. Shesha. And we'll get back to you during the panel discussion on this aspect.
I will be around and listening to other speakers, sir. Thank you so much.
Now, I take this opportunity to welcome one of the very distinguished scientists in the area of pulses in India, Dr. Harsh Nair. I've been associating with Dr. Harsh Nair over the last decade or so, and especially when we started interacting closely with Dr. Bindu Madhava, who has been working for the World Vegetable Program. In this regard, we came very closely interacting with Dr. Harsh Nair. As everyone knows, that Harsh Nair is the professor in the Punjab Agricultural University, Punjab University, Chandigarh, and his research interest has been on the plant biology, but more specifically, I would say, Dr. Harsh Nair's interest has been in the area of the pulses. As I just now mentioned about Dr. Basu, who is a synonym for the pulse programs in India, Dr. Harsh Nair actually is a synonym for the basic insight that he is providing in the area of the pulses, more specifically, the pulses reaction to the high temperature, pulses reaction to the stress situations, and pulses in terms of the potentials that they have in the yield programs and etc. As a consequence of his basic insight that he has developed over years, he has been recognized internationally, and he has probably one of the few scientists in India working in crop sciences, has developed collaborations with many international institutions like University of the Western Australia, CSRO Australia, ICRADA, Morocco, and of course in ICRISAT, along with the programs of the World Vegetable Center. He has been contributing immensely in his collaborative programs to provide greater insights, more specifically in the area of the pulses, chickpea and others, right? And he has been very productive and has been publishing very high impact journal publications and also guiding students. And he has more than 25 students completed the PhD program and his guidance. More than anything else, Dr. Nair is an excellent person and human being. And it is good that by recognizing the organizers, mainly Dr. Basu and his colleagues, rightly invited Dr. Harsh Nair to give an invited talk today on impact of high temperature on food legumes and approaches to improve the heat tolerance, which has been his area of research close to his heart. And I'm sure we're all going to be benefited with his lead lecture. Now I request Dr. Harsh Nair to give his presentation and seminar. Dr. Harsh Nair, please.
Thank you, sir. Thank you so much. I hope I'm audible. Am I audible, sir?
Yes, yes. Please go ahead, Dr. Harsh Nair.
All right. Good afternoon, everybody. Professor Uday Kumar, thanks for the kind words you have spoken about me. Highly eminent panelist in this discussion, Dr. Basu. I shall be presenting an overview of the work which I've been doing for the last few years in collaboration with several institutions. This is the outline of the presentation. Why should we be working on heat stress? What are the impacts of high temperature stress on food legumes? I'll be talking about some examples on which we are working upon. We are basically working out the threshold temperatures in different legumes, how their reproductive function is affected, what are the possible mechanisms which are related to heat tolerance. We are also looking for the opportunities how to develop heat tolerance in the legumes. So why should we be working on heat stress in legumes? Because of climate change, possibly the temperatures are rising. So this is one of the major aspects we are consideration that heat stress would be a major issue in future for the legumes, particularly for the cool season legumes and also for the warm season legumes. If we look at the data about the temperature in the night season, in the night time and in the daytime, the night temperatures are increasing more in comparison to the day temperatures, which is a major reason of concern because the warm nights are affecting the crops very badly, as per the literature reports. This is the recent information from the NASA Earth Observatory, where the temperatures are going tremendously up. So in the north part, you can see where we are placed and where IPR is placed. In the north part, the temperature is reaching 48 degrees Celsius, which is extremely detrimental even for the summer season crops. So as you can see that the heat waves, the number of states, they were 9 in the 2015, they have increased to 23 in the year 2019. And 2020 was one of the hottest year on record. So the heat waves are likely to continue and they are likely to intensify more in future. So there are some, these are some reports that the temperature not in, is rising locally more in comparison to the global average. So in Delhi, for example, a few years back, the temperature was 7 degrees above normal in the month of April, so which is not good for the summer season legumes which are cultivated after wheat. So a study from University of California has also indicated that the suicidal rate among farmers because of high temperature stress damage to the crops is increasing. So this is a reason of serious concern in future for agriculture. Another reason is that there is a large shift in the legumes from environment to the warm, short season environments. If you see this picture, the source of this information is from ICRISAT. The area under pulses in the north part is decreasing, while in the central and south states it is increasing, which are comparatively warmer. So they are likely to face more heat stress in these regions. This is one of the examples in South India, where a picture was taken in Kurnool district of Andhra Pradesh. You can see that the crops are also being grown under late sown situations. Chickpea in this region is grown from September to January. You can see here in this picture, uh, one of the side is ready to harvest, another one is close to maturity, and this has been sown afresh, so it is at seedling stage. This one is going to face heat stress. It has been sown in the month of December, or at the end of the December. It's going to face heat stress, which is likely to cause severe effects at its reproductive stage and seed filling stage. There has been introduction of short season mung bean and urd bean between rice and wheat, because there is a time gap between the two crops, so it's possible to introduce some summer season food legumes such as urd bean and mung bean. The problem is that these legumes would be facing or are facing temperatures exceeding 40 by 30 degrees Celsius at the time of reproductive stage, so which is causing a lot of damage to these crops. We are working on this aspect. So what, in summary, we are doing? We are assessing the impacts of heat stress on some cool season and warm season legumes. We are evaluating their temperature thresholds under controlled environmental conditions. We are taking core germplasm from different institutions like ICADA, IAPR, ICUSAT, World Vegetable Research Center, and we are identifying the heat tolerant genotypes. And we are also identifying what are the possible reasons of the damage at reproductive stage, and also working on some of the cellular mechanisms related to heat tolerance. So as I mentioned, we are working on chickpea, lentil, and beans. These crops have an optimum temperature. For example, for cool season crops, the optimum temperature is between 15 to 25 degrees Celsius. The temperature exceeding 32 by 20 degrees Celsius is considered as heat stress, which causes damage at the reproductive stage and seed filling stage. In case of beans, the optimum temperature is 30 to 35 degrees Celsius, and the temperatures exceeding 35 to 25 degrees by 25 degrees Celsius, they are considered as heat stress. And the response is generally the reproductive stage is more sensitive. We are there are several screening methods which we are working upon. We have chosen these methods based upon their usage in other crops. So we are working on canopy temperature depression, stay green trait, chlorophyll fluorescence, stomatal conductance, thermal imaging, membrane damage, which are leaf-based traits. We are primarily focusing on reproductive function, where we are looking into the pollen and the stigma and the ovial responses under heat stress situations. Being a physiologist and a biochemist, we are looking into the cellular mechanisms related to heat tolerance. Because of lack of time, I won't be able to present the information on all these traits, but particularly we have been more interested in the reproductive function. So I'll be showing you some quantitative data as well as pictures on this aspect in different legumes. So we are mainly focusing on chickpea, lentil, and beans. I'll be showing information from 3 crops here, crop-wise information starting from mung bean. Now, how to impose heat stress? There are 3 ways to impose heat stress. One is that we can sow the crops late than the conventional sowing. As a result of that, the crops will face heat stress during reproductive stage and the subsequent stages. If we have access to hot locations, we can grow them there also. We are also focusing on control environments where we have walk-in growth chambers and controlling the temperatures in walk-in growth chambers and exposing them to varying temperatures to find out the thresholds. So it's easy to handle the crops under controlled conditions, because while handling heat stress situation, irrigation is very important. Otherwise, we get the confounding effects of the drought stress. So it's easy for us to handle the plants in the pots and as well as to take the observations. So this is the temperature profile during normal sown and late sown situations. When we exceed the conventional rate of sowing, we can get the temperatures which are stressful, that is exceeding 40 degrees Celsius. The profile is shown here. Under normal sown situation, say we get 40 degree The range is 33 to 40 degrees Celsius, while the night temperatures are 21 to 26 degrees Celsius. Under late zone situations, we can get a rise in the temperatures, 37 to 45 degrees Celsius day temperature, and so is true for the night temperature, where the temperatures can go up by 2 to 3 degrees Celsius, which gives us the opportunity to expose the plants to heat stress. So these are this is some representative data from particular genotypes, where we can see what happens in case of, say, for example, mung bean. We can see that the flower number is not affected significantly, but the pod set goes down tremendously. As a result of that, the pods per plant, they can go down by 43%, seeds by 45%, seed weight per plant can go by 48%. The information can vary, the data can vary depending upon the genotypes. Some of the visual symptoms which we can record: scorching of the leaves, abortion of the flowers, abortion of the pods, and the pods are very badly shriveled. We are primarily into reproductive function, so we have been collecting the flowers at the time of reproductive growth and understanding what happens to the pollen viability, which becomes very poor. Pollen germination has come down to just 11% compared to 75% in the control plants. And looking at the scanning electron micrograph pictures, the pollen morphology is very badly affected. They are very, very badly shriveled. Looking into the stigma receptivity, which is an easy test to measure what happens to the pollen load as well as stigma receptivity, we can see that there is a problem in anthrodescence. Because of that, the pollen load is not apparent here. In this case, if we look at the scanning electron micrography pictures, we can't see the pollen load on the stigma surface. We can see it in the control plants very easily on the stigma surface. So all these events are resulting in poor pollination, presence of less pollen grains, and as well as, if we follow the pollen tube growth using fluorescent microscopy, we can see the pollen tube in the control plants, but not in case of the stress plants, which disrupts the fertilization, leading to the abortion of the pods. We have been screening a lot of material from World Vegetable Center, especially taken from Dr. Ramakrishnan Nair and Dr. Bindu Madhva, and we have been giving information to them. And this is the pod situation in selective genotypes, where you can see healthy pods under normal zone situations, while under heat stress situation, they are already badly affected. So we have been screening the genotypes based upon their pod appearance and many other traits also. We are also working on the thresholds to find out what happens at different temperatures to the reproductive function. For example, pollen germination starts getting affected after 35 by 25 degrees Celsius day and night temperature. After that, there is a decrease. Stigma receptivity also starts affected at the same temperature, but you can see that the rate of decrease is more in the pollen germination. Pollen is more sensitive than the female components. Many studies have indicated in our investigations also, and in other situations also. But at the same time, the female components are also playing a very important role. This corresponds with the number of pods which are produced at the corresponding temperature. You can see that after 35 by 25 degrees Celsius, the seed number starts getting down. At the same time, the seed filling is less affected. So seed filling process is comparatively more resistant to the heat stress in comparison to the pod number as well as the seed number. We have been comparing the effects of increasing night temperatures on the left side, increasing day temperatures as well as night temperatures on the right side. You can see here in the night situation, if the temperatures increase from 23 degrees Celsius, the rate of decrease is very fast in comparison to the increase in day temperatures. So this means that night temperatures will be more detrimental. The increase in night temperatures would be more detrimental for reducing the number of pods and number of seeds. In a similar way, we are working on the chickpea, where we are exposing the plants to late sowing situations as well as in controlled environments. Whenever we grow the chickpea under late sowing situation, after this arrow, you can see that the temperatures can exceed 30 degrees Celsius, can reach 35 degrees Celsius and more during seed filling stages. So which gives us a good opportunity to find out what is happening under heat stress situations. It's easy to control the plants under control environment. I have told you the reason, because of irrigation requirements. So in general, there is a severe reduction under heat stress situations. There is damage to the pods on the right side, you can see, because of high temperature stress. Damage to the pollen load with less pollen on the stigma surface because of heat stress. the pollen germination and the viability is affected severely. So is true for the stigma receptivity. At the same time, we have been following the track of pollen tube also. So you can see here on the normal situation, the pollen tube is able to reach the ovules, resulting in fertilization. But at the same time, in case of the heat stress plants, there is no pollen tube here, so the ovules get aborted. We have been quantifying this information in different kinds of genotypes, in tolerant ones and sensitive ones also. And recording the information on reproductive biology, you can see the difference here. The black bar represents the impact of heat stress in sensitive genotypes. Below bars, below graphs, you can see there is a tremendous reduction, particularly in the pollen germination and in the pollen load in case of sensitive genotypes in comparison to the tolerant genotypes, but both are affected at the same time. We have been quantifying the impacts on the growth as well as on the yield contributing traits also. If in nutshell, I have to tell you that the number of fill pods is impacted to a very high extent in case of sensitive genotypes. 55% reduction can be seen in comparison to the tolerant genotypes, where it's around 12%. At the same time, seed number is going down, seed weight is going down by 66% in sensitive genotypes in comparison to the tolerant genotypes, showing around 39% reduction. Working on the thresholds, we have found that the pollen germination is sensitive at 35 degrees Celsius and above that. And in the tolerant genotypes, they are able to face 35 degrees Celsius in comparison to the susceptible genotypes, which start showing reduction even at 30 degrees Celsius. So in a similar way, this is the response of some tolerant and sensitive genotypes. The tolerant genotypes are able to perform better in comparison to the sensitive genotypes at high temperatures in terms of pod number as well as the seed yield. Now, why we are choosing contrasting genotypes? Because as a physiologist, we have been working on some traits which can yield us some information to find out what are the possible mechanisms or contributing factors related to the heat stress impacts. One of the mechanisms on which we have been focusing is on sucrose metabolism. So there, on the right side, you can see some of the components of sucrose metabolism involving the enzymes and the transporters, which are downregulated in case of heat stress situations. So sucrose is basically a product of photosynthesis, which is going to the flowers and the pods to enable them to maintain the reproductive function and the seed filling situations. Whenever there is a disruption in the sucrose metabolism, it's likely to impact their function as well as the growth. So what we have been doing is, this is just a small data from a contrasting study where we have been taking few tolerant genotypes and few sensitive genotypes, exposing them to normal situations and under heat stress situations, and working out the sucrose concentration in anthers, in pollens, and the other enzymes which are related to this metabolism. So we have pinpointed that there is severe reduction in the sucrose concentration in the anthers as well as in the pollen grains, especially in case of the sensitive genotypes, which is related to the synthesis of sucrose as indicated by reduction in the activity of enzyme sucrose synthase, which is going severely down in case of sensitive genotypes in comparison to the tolerant genotypes in case of anthers as well as in pollen grains. At the same time, the enzyme acid invertase, which is required for the utilization of the sucrose, which is also going severely down in case of sensitive genotypes to a higher extent than the tolerant genotypes. So we speculated that one of the possible reasons which can cause abortion or poor performance of the pollen grains is related to poor concentration of the sucrose or interruption in the sucrose synthesis. In case of lentil, this is the third situation. In lentil also, we are working in a similar way, exposing the plants to late zone situations as well as under controlled environments, where we can raise the temperatures to stressful situations and then examine the plants under that. So there is a considerable reduction under heat stress situation in the lentil plants also. Various kind of symptoms which can be seen is sclerosis, clustering of the leaves, curling of the leaves. At the same time, there is abortion of the flowers and the pods, which is visible more clearly in the next slide. You can see there's a bad effect on the flowers also, and the pods also. So lentil has been found to be comparatively much more sensitive to heat stress at the same temperature in comparison to chickpea. These are more close-ups where pods are shown to be damaged by the heat stress. Flowers are badly impacted. The seeds size is very, very poorly affected, which also impacts the quality of the lentil seeds. We have been working on some ultrastructural studies to find out what is happening to the leaves. This is in addition to the photosynthesis and other components which we have been doing. I'm just presenting some salient information which have pictures to convince you what is happening here. In comparison to the control plants, you can see a severe damage to the chloroplast as well as to the nucleus. Trichomes have a very important role in conserving water, and stomata also have a same role in this situation. So what we have been doing, we are comparing a heat tolerant genotype and a heat sensitive genotype under heat stress situations. What we have found out, heat tolerant genotypes are able to maintain more trichomes on their leaf surfaces, but less stomata. It's surprising, but at the same time, heat sensitive genotypes are able to maintain more stomata, but very few trichomes on their leaf surfaces. So this is probably related to their adaptive features. Looking at the reproductive function, as we have done in other crops also, there is severe reduction in the pollen performance. Pollen germination is poor, pollen load is not good, stigma receptivity goes down, ovule size as well as the viability goes down. The close-ups under scanning electron micrography can clearly tell you that under heat stress and compared to the control plants, the pollen are shriveled, the anthers are very badly damaged. In a similar way, we have been tracking the pollen tube as for the other crops also. There is no pollen tube, or it is disrupted in the styler region, not reaching the ovules. We have been quantifying this information on the reproductive function related traits. You can see lentil is very badly affected in comparison to the chickpea, which are both cool season crops. Viability is severely affected. Germination, load, stigma receptivity, ovule viability, all are quite severely affected under heat stress situations, which are the possible reasons of poor performance of the flowers. We have been quantifying the effects on yield traits. You can see here the yield, seed number of seeds can go down by 90%, seed weight can go by 83%, seed weight by 50%, and harvest index 69%. So looking at the threshold, we have seen that in comparison to chickpea, the pollen germination is affected earlier at 30 by 10 degrees Celsius in case of lentil, and so is true for the stigma receptivity. In this case also, we have seen that the rate of decrease as we increase the temperature, it's more in case of pollen germination in comparison to the stigma receptivity. So this matches with the yield traits. You can see here at 30 by 10 degrees Celsius, there is a reduction in the fill pods. Then at the same time, there is a reduction in the single seed weight, but it is comparatively less affected in comparison to the fill pods. So we have been scanning the seed surface patterns to find out what is happening at the finer details. This part is for the tolerant genotypes. This part is for the sensitive genotypes. You can see this is the control situation. These are papilla on the seed surface, just to find out what happens to the seed actually, not at the functional level, but at the structural level also. The heat tolerant genotypes are also impacted because of heat stress. This indicates poor development under heat stress situation, but heat sensitive genotypes are very badly affected. So we have been testing these seeds for their germination as well as quality impact. So we have been finding severe reduction in the vigor related traits. This is one information which we published a few years back to show you that the accumulation reserves, starch, proteins, sugars, fats, and other patterns are also getting badly damaged as the heat stress is increased. This is just for one graph. We can see here this control plants for a heat tolerant genotype, the heat stress plants for a heat tolerant genotype. This is control plants for a heat sensitive genotype, heat stress plant for a heat stress plants for a heat sensitive genotypes. You can see severe reduction to a more extent in case of sensitive genotypes. We have been comparing chickpea and lentil at different temperatures under control conditions, and we have found out that at earlier growth, when we are exposing the plants to the same temperatures, lentil is more badly affected in comparison to the chickpea. So we have done further experiments, also not able to present the entire information here, but it has been published recently in Journal of Agronomy and Crop Science. This is to show you that the gray bar shows that there is more reduction in the lentil income for the pods number per plant, seed weight per plant, when we are exposing it to the same high temperature stress. So what should we do now? So after the impacts, I'm going to show you what can be done now to develop heat tolerance. So we have two ways where we can explore the germplasm and screen it for heat tolerance, and then make crosses and all that. And at the same time, look for the wild relatives, whether they are carrying some heat tolerance or not. So this is a trial from the ICRISAT where they have extended the growing season by at least 2 months. So after that, this is the situation. In normal zone plants, the reproductive stage, the temperature is around 30 degrees Celsius or less than that. And in a late zone situation, the temperature can be higher than 30 degrees Celsius, reaching 35 degrees Celsius. Under these situations, they have found large genetic variation in a set of 300 genotypes. And on that basis, I want to show you that we have also been doing these experiments under controlled conditions. You can see that there is tremendous difference in the germplasm, in the growth, also in the pod production, when we are exposing them to controlled conditions. This is a study from the ICRISAT where they have been able to find out some of the best genotypes which can produce a good number of pods, and these candidates, genotypes have been picked up and further evaluated at many locations. You can see here it's a list of chickpea genotypes, and out of that, one of the promising line is ICCV 92944. I think all the legume scientists, they know that this has been released after screening as a variety, and it has shown promising results in the field conditions where you can see its comparison with a sensitive genotype. It's going very well. Many trials have been done in different states, and it is able to do well under heat stress situation. On the right side, you can see this plant. And as a result of that, this line has been released in 3 countries, Myanmar, India, under different names. And in India, it has been released as JG14 as a heat tolerant variety. And its trials in Uttar Pradesh and in Madhya Pradesh, they have shown 25 to 30% increase in the yield in comparison to the local cultivars. So this is one of a success story coming out of the screening results. And surprisingly, this line is maintaining superior pollen functions. As a result of that, it's able to set more pods. So pollen germination can be taken as one of the best criteria for choosing heat-tolerant genotypes. These are further studies done in ICRISAT where they have been molecular mapping the genes and the QTLs. They have made crosses with the tolerant and sensitive genotypes. They have done phenotyping also based upon these traits, and they have found that there is a twofold difference in the RILs under heat stress environments in comparison to the non-heat stress environments. Subsequently, QTLs have been identified for heat tolerant genes, which are related to percentage pod setting, number of fill pods per plant, and other yield traits. And it has been suggested in this study that these QTLs can be introgressed through marker-assisted breeding for heat tolerance. This is also a study from ICRISAT where they have been working on proteomics. This is, you can see, this is in this study, normally you can identify a number of proteins which are expressed in relation to heat stress. In heat tolerant genotypes, they have identified 482 proteins which are related to different categories. On right side, you can see that they have suggested the mechanism which is commonly expected after this kind of a study, that heat stress tolerance is related to accumulation of osmoprotectants, accumulation of some secondary metabolites, stability of the membranes, so activation of different kinds of antioxidants, and different kinds of hormones. In genomics, some transcription factors have been identified, which could be possible candidate genes for heat tolerance. Some QTLs, which I mentioned, they have been identified for heat tolerance. At the same time, some candidate genes by Varshney group have been identified, which will be providing more information after the validation. This is what we have been doing, and ICADA has been doing, IIPR has been doing. This is a study from ICADA where a technique-focused identification of germplasm strategy FIGs have been developed by Dr. Shiv Kumar and his group, and they have been doing screening using subsets of certain lentil genotypes and focusing on few criteria. On that basis, they have been able to identify some heat-tolerant genotypes. In a similar way, Some screening studies have been done, and a few heat-tolerant genotypes have been identified. The study has been published by Dr. Shiv Kumar group recently. And interestingly, here also I want to mention that pollen germination has been positively found to be correlated with the heat tolerance. We have also been doing at our place similar kind of screening studies. This is a material procured from ICADA, IAPR, where we had done the screening and identified some of the heat tolerant genotypes. So these are the possible lentil genotypes, which the information is all the time being passed on to these institutions. Just to show you what we have been doing in the contrasting genotypes to show you very clearly that heat tolerant and heat sensitive genotypes, they are very, very clearly different from each other when they're exposed to specific temperatures. So we are looking into some contrasting genotypes to identify the traits and the mechanisms. So on the left side, you can say there are 5 heat tolerant genotypes. On the right side, you can say heat sensitive genotypes. And there is tremendous reduction in the ports number under heat stress situation with heat sensitive genotypes in comparison to the heat tolerant ones. We have been correlating them with the different reproductive functions as well as leaf functions. We have seen that the pollen viability is quite poor in case of heat sensitive genotypes in comparison to the heat tolerant genotypes, which are showing more viable pollen. At the same time, pollen germination is high in heat stress, in heat tolerant genotypes under heat stress situations in comparison to the heat sensitive genotypes. Looking at the leaf function, just one of the traits I'm showing here, membrane damage is taken as one of the highly reliable traits by plant physiologists, and it has been suggested to the breeders also to take up this for screening purposes. It has clearly defined that the heat sensitive genotypes, they have more sensitive membranes, showing no damage to their membranes in comparison to the heat sensitive genotypes. In a similar way, we have been working on mung bean, screening them at our place, and identifying the material taken from World Vegetable Center. Recently, we have collaborated with the IPR, and they have started providing us the lines, and we are screening them and finding out the promising genotypes. This will be the last slides before conclusions. We are yet to work on wild genotypes. We'll be starting this work now and trying to identify whether we can use some hybrids to identify or to generate some basic information related to heat tolerance in chickpea, lentil, and other crops. So this is just a list of the different kinds of wild genotypes which are available for our study. So what our study has shown so far. Heat stress is going to cause serious problems for the legumes, but fortunately, we have large genetic variations in the existing germplasm, which would provide us sources of heat tolerance. And at our level, we have been working on the different traits based upon leaf and pollen, so as to identify and give it to the breeders for this large screening strategies. We are working on the different mechanisms. Just as a physiologist, I'm more interested to find out how the plants are getting heat tolerant. So we are working on different aspects, which include photosynthesis, which includes respiration, nitrogen fixation, and antioxidants. The information are not presented here, besides some of the non-destructive traits required for screening of these genotypes. It's very important to study the impact of heat stress with other stresses also. We have been working on chickpea and lentil. We have published also, and in drought and heat interaction, this is available in the literature is available. We have started now looking into the wild relatives, and we are into the attempt that if we can develop some effective screening techniques for these kind of studies. These are the key references where we have published some of the useful studies. Anybody who is interested to find out what we have done so far, what is the summary of the other work from different labs can see this literature. These are one of the most recently published work, which is on chickpea as well as on the contrasting genotypes, what kind of traits we can use to identify the heat tolerant genotypes. So plant growth hormones, they have a strong potential because there are many studies which have indicated that some of the molecules such as salicylic acid and gamma aminobutyric acid and some of the osmoprotectants, as well as racinosteroids, they have been found to be very effective in inducing heat tolerance. This is one of the shortcuts to induce heat tolerance, which we also have been trying, and we have found some success in this case. So at the end, I'm extremely thankful to my students, my collaborators, and all the people who have been involved in the study, providing me germplasm and helping me from time to time. So I'm thankful to the organizers for providing me this opportunity. Thank you so much. Over to you, sir.
Thank you very much, Dr. Schneier, for a very comprehensive and excellent insight that you have provided with regards to the heat stress response of the two important legumes, that is Caesar as well as in the lentil. And of course, as you rightly said, you have taken to provide some basic information on the most critical stage. That's where the most interesting point is, the reproductive structures are concerned, right? And of course, we'll come back at the time of the discussion later to get some more idea about what really the projection that you're going to have in terms of the selection indices, in terms of receptivity, pollen viability, and probably a few other, and the technologies that you have developed to quantify genetic variability in these particular traits, and also to identify genotype differing in this aspect. We'll come back and for the discussion. Look forward to hear you again based on the inputs and the queries that comes from the panelists and others. Thank you very much. An excellent presentation, Dr. Harsh Nair, who has given some good insight to us. Now, gentlemen, we move on to the third presentation. That is from...
Hello, Professor Uday Kumar.
Yeah.
Hello, can you hear me?
I can hear you. Yeah.
Our organizing secretary want to make an announcement, urgent announcement. After that, we'll just start the session.
Sure, sure.
Yeah, sorry, sir, for the interruption. There's an urgent announcement that at 3:45 sharp, DG ICARDA, Dr. Ali Abu Sabah will be connecting for 15 minutes for his address for the Pulse Webcon.
Okay.
So, so he will be connecting at 3:45 sharp. So before that, we can have the presentation of Dr. Madan Pal, and then, sir, we will be continuing with the address of DG ICARDA, Dr. Ali Abu Sabah.
At 3:45, 3:40.
Yes.
Yeah, we'll do that. I think, Gaushia? Now, shall we go ahead and for the presentation of Dr. Madan Pal? I don't have to introduce Dr. Madan Pal more specifically. Madan Pal is presently the professor in the Department of Plant Physiology, IRI, Primary Institute, and one of the strong... Hello. Can you hear me?
Yes. Yeah, yes, you are audible, sir.
But I can't see you. That is the thing.
I'm here, sir.
Yeah. Dr. Madan Pal has been working in the area of the climate change. Always we call him the moment. We remember about Madan Pal. He is a man with the climate change who has worked constantly with the environmental effect assessment panel as a member and also as a reviewer. And in addition to that, he has been working in the climate change scenario in terms of the effect of elevated CO2 and temperature on plant growth and the development in this direction. And he has been visiting, and he visited a number of countries and got the interaction with international scientists in this very important topic of how the plant respond to the changing environment, both in terms of a combined CO2 and high temperature aspect is concerned. I remember vividly, he used to develop the heat channels and several different what do you call it as the phenotyping technologies to study the effect of high temperature and the high CO2 in different crops about it, right? And he has been awarded the UK-India International Fellowship by Royal Society, and he is a recipient of the Chennai Gold Medal. And he has become more or less synonym to the Indian Society of Plant Physiology. The moment we remember about the Physiology Society, it is Madan Pal, who has been very ably participating and as a secretary and also as a treasurer for several years, and enthusing the many people to get into the very exciting area of plant physiology, where we look into how actually the plant grow than how to grow a plant. We look forward to hear Dr. Madan Pal today on a topic, phenotyping the chickpea for drought and high temperature tolerance, right? As the organizers just now has announced, you please continue the presentation and we'll have a short break at 4:45 for 15 minutes, and subsequently we continue our presentation. And this component, I'm sure Dr. Basu or the organizers will monitor the moment the time comes to 3:45. Until that, the podium is yours, and I request you to present your presentation, please. Dr. Madan Pal, please.
Most respected Professor Uday Kumar, my esteemed teacher, mentor, as well as guide of our research work established at IRI. It is his kind guidance and support that we have been able to establish most of the advanced high throughput research facility, particularly the Phenomics platform. It is the result of his hard work and support of Dr. Uday Kumar sir. Thank you very much, sir, for your concern. I am really happy that I am presenting some of my work of the chickpea when you are the mentor of this session. So as I have seen my predecessors, Dr. Sesha and Dr. Harsh Nair, they have already spoken different aspect of the pulse crop, and Sesha has already given the basic ideas of the mechanism, etc., everything, or the physiology of the pulse actually he has covered almost. And so also the Harsh Nair has spoken about the various mechanisms, tolerance mechanisms involved in the pulse crop in general, but simply my presentation here is that basically what our experience and particularly the methodologies which we have developed to work on the heat tolerance mechanism, not the mechanism basically, but the phenotyping, particularly in chickpea genotypes. And basically this work, it was one of our commitment. So I'm not, I have worked for 4 or 5 years on this crop. After that, I have been basically focusing on the rice crop. But this was the work where we had this commitment of the chickpea crop in our NICRA program. So I want to share you some of the work which we have conducted at IRI.
Screen point.
So regarding the chickpea and stress resilience, since we are talking about the high temperature stress, we know that this crop is a most hardy crop and it can thrive well under the most limited conditions. But the temperature is a major environmental factor which can affect particularly the timing of the flowering is very sensitive in chickpea crop for the high temperature. And the optimum range of this crop is, as already mentioned, it is 15 to 25 degrees Celsius. So if the temperature goes below 15 degrees Celsius, it can also affect the growth and development, particularly at the vegetative stage. On the other hand, the temperature exceeded 25 degrees Celsius, it gives the harmful effect on the reproductive developments. It has been found that when the high day and night temperature goes more than 30 or 16 degree, it may affect the crop yield because of the various constraints on the development of the reproductive structures. So as such, the crop is very tolerable for the temperature as well as the drought stress conditions. But when we look into the different projections in the climate scenario, when the temperature can further increase and also the rainfall pattern is expected to change, we need to understand, we need to phenotype the genotypes which can basically be tolerant to the high temperature stress environment. So we started this first work because, as Dr. Harsh Nair also mentioned, the phenotyping of high temperature tolerance, particularly when we have the large number of genotypes, so the staggered sowing or delayed sowing is the best alternate, where we can have the crop, the delayed crop can have can experience the high temperature, particularly we are interested in the seed pod filling stage. So keeping into that one, we have taken nearly more than 30 varieties we were grown. And this is the work we conducted in the NICRA program in the year 2013. So we had the 2 dates of sowing, as I mentioned, through the staggered sowing. The first one was November 10th. Then it was delayed further by more than 1 month. So 15th December was the next date of sowing. And these are temperature regimes for the whole crop season, which we recorded here. So this is the important part to understand here. When we look into temperature change at different phenological stages, it is very important. So what we observed that the S1 at both the date of sowing, we measured the temperature at the 60 days after sowing, that's the vegetative stage we considered. So what is the main effect was there, that the delayed sowing crop was basically experiencing the low temperature, less than 15 degree, and the night temperature was also nearly below 5 degrees Celsius. On the other hand, the another important effect was on this delayed sowing was that the pod by 50% podding stage as well as the pod filling stage, it was having it experienced the temperature more than 30 degrees Celsius. And as I mentioned earlier, that more than 30 degree temperature can be considered little bit detrimental for this crop, chickpea crop. So this was just the screening of the crops genotypes at the pod filling stage in both of them. So we have just a representative picture of the genotypes. Just you can see the temperature did not find much change in the thermal imaging tissue temperature. In the normal sowing, it was 23 to 28, and also slightly lower in the delayed sowing. So as such, the significant differences were not there because of the delayed sowing in the tissue temperature. And then this is the result on the biomass production and the grain weight of the genotypes. What we found that there was a great variability in terms of the biomass accumulation as well as the total grain weight per meter square, which was recorded in all the genotypes. And then some of the genotypes, they showed very small reduction compared to the other ones. So based on the response of this one, we found that, and that this was the percentage in case. So what we observed here is that percent reduction showed that late planting showed about 9 to 67% reduction in total biomass. On the other hand, the same genotype, we found 8 to 64% reduction seed yield. So you can see the large variation is there among the percent reduction, and it was the one of the reason what we expected that the total biomass, because of low temperature, delayed sowing crop basically it experienced the low temperature at the vegetative stage, and one of the reason can be there that the vegetative growth is hampered and biomass production may be affected. But even then, that the genotype which showed less than 10% reduction in biomass of the total, maybe the effect of low temperature or maybe the high temperature regime at the pod filling stage. So we can consider them as the tolerant type. So based on this one, it was the susceptibility index was calculated, and among them we found that the 4 of them, when we have the susceptibility index less than 0.5, then we can consider them they are having the as a tolerant one. So we have identified the 4 of them as one of the can be considered the donor, and one of them is JG11 also. What Dr. Nair has shown, JG14 was there. I think it's the same germplasm representing here. So this was the region. So because in the staggered sowing, what we experienced, the temperatures low as well as there was a large variability. So what we thought that why don't we grow the time we sow this crop in the field at one season continuously, and we expose them to the high temperature. So when we have taken in this experiment was conducted in the next season, and we have grown only the 2 genotypes in the field conditions with 5 replicates of plot size 6 into 3 meters square. So what then we in the same genotype, the same stage, we have fixed these polychambers were fixed with the help of our agriculture engineers, and the idea was that because of the this poly transparency, the heat, the light conditions will not be affected, but the temperature can be increased. So what we observed that these polychambers were fixed at the floral initiation stage in the field conditions, and the data loggers were fixed inside the chambers. And what we observed from the data logger, the 3.5 degree increase in the temperature was there below the polychambers, and the humidity also increased by 5 to 7% as the average in all the polychambers. So the idea was there, then the crop can be grown in the same time, so there can be no differentiation of the effect of the low temperature, and at the high temperature can be there. So that the open plot was the control, and the high treatment temperature was considered below the plants grown into the inside the below the chambers there. So what we observed that in general the plants which covered, you can see from the picture, the lower picture also, the plants which were grown within the inside the polychambers, in this were the plant matured 11 days before the control. That was the major visible changes were observed here. Then also we have recorded this data on the yield rates also, and what we observed, the shoot biomass as such in both the genotypes, because of the high temperature of 3.4 degrees, there was no effect there, but the total seed weight was marginally decreased there. So on the basis of this one also, you can see the 100 seed weight as well as the harvest index, there is no much significant changes were there. So this study concludes that the chickpea crop is adapted per se to the high degree temperature up to 4 degrees higher than the ambient one. So what we can conclude from here that as the projections are there in the climate change scenario, at least because we have grown in the same environment and the temperature increase was the same conditions, so at least we can presume that the temperature projections are there up to 4 degrees in next 10 years, 20 years. So this can be actually taken as the one of the information that high degree temperature, 4 degree higher temperature up to 4 degrees, the crop can tolerate it. Then this is the same one. So then another, the same genotypes have grown, and then because the level of temperature, we were not able to increase more than 4 degrees. That was the one of the idea that whether it is 4 degree up to 4 degree, these genotypes showed to the tolerance. So we wanted to go for the high temperature, that up to what level these two genotypes can have the tolerance level. So another study was planned with the same genotypes, and we have grown them under the polytunnel conditions when the temperature was increased to about 5.93 average temperature, particularly at the flowering and the protein development stage. And what we did in this is polytunnel study, the same genotype, we have selected the 15 genotype from both the each genotype, and they were tagged before the flowering and the podding, and the number of pods of flowers produced per day were counted every alternate day. Then temperature was also recorded in the same way in the polytunnel by using data loggers. So what you can see, these are the data on the number of flowers produced at different days, you can see. So we started from the 80 days and continued up to the 114 and 18 days was there. So this is the dotted lines are both the genotypes. Dotted lines showed the number of flowers produced under the normal temperature conditions, while the open conditions rather, while the dashed line, they are showing the number of flowers produced by both the genotypes under the high temperature conditions. So two important messages from here we got that under the high temperature conditions, we get the two flushes of the flower. The first was almost false flush of the flowers was there. And as such, when we compared the total number of flowers produced in both the genotypes, under control conditions, they were higher than the high temperature exposed. Same way, the lower graph showed the number of pods produced by both the genotype. Same way, the number of pods were also lower under the high temperature compared to the normal conditions. And another important thing what we observed here is that under high temperature, the flower initiation was observed about 12 days earlier in both the genotypes compared to 90 days, the days after sowing under the normal conditions, it occurs at 80 days. So then this is the data on the number of flowers and pods you can see in both the genotypes. Because of temperature stress, it was showing the reduction is there, but they show the variation because in one of the genotypes reduced by 14%, while in 1105 it reduced by 5% was there. So unlike the flower production, high temperature also significantly decreased the number of pods. It was 29% and 34% in both the genotypes. Then as a result, we calculate the flower abortion ratio. It was also decreased because of the high temperature. Then this is the biomass partitioning. What we observed in both the genotypes, if you look into the biomass at the control conditions, in 1053, it was 45% in the pods. On the other hand, in the high temperature conditions, it decreased to 30%. Same way in 1105 also decreased from 35 to 29%. And the increase was in the stem side. You can see the stem 27% was there, but at 47%. So it shows that this high temperature exposure at the reproductive stage particularly can also affect the biomass partitioning in the genotypes. This is the data on the total biomass and the yield in both the genotypes. And now another study we have planned to, because the temperature initially it was 3 degree hot, then it was 4 degree, and we have studied up the 6 degree. A 6 degree temperature was really found to be very detrimental to affect the number of flowers as well as number of pods produced by the plants. Then another study we wanted to go, it is a control environment study again. The plants were grown with the same two genotypes in the growth chambers, and we wanted to see whether our calcium application, if we increase the cytosolic calcium by just having the foliar spray of the calcium chloride, if we are increasing the level of the cytosol, see whether it can be able to induce some sort of tolerance through the induction of the antioxidant system and the molecular response. So we have taken the same two genotypes grown under the growth chambers, particularly in the pots. So as such, the growth of the plants was not very appreciable compared to the field-grown conditions, but high temperature at the vegetative stage reduced the growth biomass. And also when we had given the application of this calcium chloride, that the growth was slightly improved. Same response was observed in the another genotype. So it shows that at the vegetative stage and the temperature level here, it was at 10 degree higher than the normal. So at 10 degree temperature, basically it is really very, it can be very detrimental for growth stage also. So at vegetative stage, it has affected the growth also. And when we have the calcium application, it was able to improve it. Then, sir, this is the response at the flowering stage. What we observed same way, it was not very appreciable, but application of calcium at the before exposure high temperature also was able to show some improvement. Then what we observed, this is the major effect was there through the lipid oxidation was basically lower with the application of the calcium in high temperature stress, and there was induction in the activity of both the important antioxidant enzymes like the ascorbic oxidase and the SOD. And this is the data also. You can see the shoot biomass was not affected, but if you look into the total seed weight in both the genotypes showed nearly 50% reduction. But when we had given the calcium treatment as well as high temperature stress, the reduction was nearly 30%. So it was able to compensate about 50% loss of the heat stress in both the genotypes. So this is the conclusion from this study, what we observed up from the high temperature stress, that low temperature stress at vegetative stage may affect the biomass accumulation and is associated with the lower grain yield. High temperature may be a key constraint for the reproductive development and the yield of chickpea and the crop compared to the vegetative growth. Then high temperature projection, the climate scenario may not affect the chickpea productivity as such, rather than it can be useful for the cold stress environment, because at 4 degrees Celsius temperature under the field condition, we do not find the much significant impact of the high temperature stress. So another just a small study for the phenotype of the root traits in chickpea. Then we had taken one population, and this was because root density is one of the important trait. So we have used this technique again, following the methodology adopted by Department of Agriculture, US Bangalore. They have grown the elevated plot, and the plots, plot was fabricated by them. But here I have grown them in the black polythene chambers where the 20 kg soil was taken. It was mixed, but 40% was sand and 60% was the soil and FYM. And we have grown these plants of the population and the parent in up to the 50 days. At 50 days, we have harvested, and then we are seeing the root and density. You can see in most of the genotypes, it was increasing in the root and density due to the high stress, due to the drought stress, and some of the genotypes showed a very high increase in the root and density. Same with the root ratio was also increased. So, and this is just the last slide only, phenotyping chickpea genotype, and the field conditions also. We have taken 30 chickpea genotypes, and based on the drought susceptibility index, we have taken them. One of the donor was there, other lines like L550, Pusa Green 112, and some of the ICC line were also produce better under stress, and they can also be characterized further for their mechanism of tolerance of the stress. So, sir, I completed within 15 minutes. I hope that it will not be clashing with the lecture, sir, and discussion we can have.
Thank you, Dr. Madan Pal. I will request the Professor Uday Kumar kindly allow us to connect with Ali Abu Sawa, DG, Director General ICARDA from Egypt. And meanwhile, you can take it as a part of the sessions. And after 15 minutes slot, we'll again reconnect with you. And I will invite our Director NPC. वो तो बोल रहा है जयंत प्रति.
Hello. Good evening, sir.
Yeah.
Dr. Uday Kumar ji.
Yeah.
Namaskar.
Namaskar,
Namaskar, sir.
Yes,
Namaskar.
Sir.
Dr. Madan Pal ji. Namaskar. NP Singh, sir.
How are you, sir?
Fine, sir. Fine. Sorry for 5 minutes interruption, so that he joining from the Egypt,
Sure.
DG card. And this will be the part of the session, and then again we can connect.
Absolutely.
He will be just addressing, yeah.
Sure. We'll wait for his talk.
Yeah, sir.
Okay, well, thank you so much for the opportunity to address this very distinguished group of experts on this particular area. And also for associating ICARDA with this Pulse Webcon 2021 to commemorate the World Pulse Day being organized in India. It's been an immense pleasure for me to participate in this event, which is organized by the Indian Society of Pulses Research and Development and ICAR, Indian Institute of Pulses Research, Kanpur in India. I'm thankful to the organizers and the Indian Council of Agricultural Research, Government of India, most particularly to the Director General of ICAR, Dr. Mohapatra, and his team. It is certainly a great pleasure for me and ICAR to speak to this very distinguished group of people. As you are aware, we've observed the International Year of Pulses in 2016 in Morocco, under the UN patronage, attended by global pulse scientists, donors, policymakers, development actors, farmers, regional fora, and international organizations like FAO, World Bank, and IFAD. The global pulse research and development, progress, opportunities, and constraints were elaborately discussed, and a number of recommendations were made, much of which have been implemented so far. I am sure that, as many of you are aware, I'm sure as many of such recommendations have been addressed to develop and deploy innovations and new technologies in the form of varietal development and improved crop husbandry that reflected in farmers' fields, which now led to the production of around 80 million tons of pulses globally. Among major pulses growing countries, India is the largest producer, consumer, and importer. The South Asia region is the major consumer of pulses, but a substantial deficit is prevailing there. And countries import from international sources at a high cost of hard-earned foreign currency to cover the substantial deficit. I am glad to learn that in recent past, India and the region have made a quantum jump in pulses production, and with a very high level of ambition set by Dr. Mohapatra this morning at the level of 24 million, I think a lot more work is needed, and with the support of ICARDA and others, we are very confident we'll get there. ICARDA is among other centers, CGIAR centers like ICRISAT, SIAD, and IITA, who are engaged in pulses research and development to support farmers and consumers, more specifically in sub-Saharan Africa and South Asia. We have a global mandate for lentil, kabuli chickpea, in collaboration with ICRISAT, grass pea, and fava bean as well. ICARDA collaborates with over 40 developing and developed countries, including Advanced Research Institute. Our gene bank holds around 40,000 pulses germplasm comprising of cultivated and wild species collected from the Fertile Crescent of the Near East, all of which contribute to the advancement of production of pulses in India, including climate resilient production systems. Almost 300 varieties of lentil, kabuli chickpea, fava bean, and grass pea have been released globally, emanated from the ICARDA supplied genetic material. We would be glad to know that with the strong support from ICAR and the government of India, ICARDA has strengthened its research collaboration by establishing the research platform at Amlaha in the state of Madhya Pradesh. The research platform is engaged in research to support and facilitate pulses programs in India, South Asia, and even globally. Promising genetic material and agro-technologies are being shared with ICAR institutions and the state agricultural universities, as well as shared with national programs of South Asia through regional networks. Under one CGIAR, there is a global strategy on protein that is currently being developed, and pulses will have a key area as part of the future diets across the globe. Hence, the efforts of India in this space will be an integral part and definitely inform the formulation of this strategy. Pulses are known as house of nutrients, as they are a vital source of protein, macronutrients, vitamins, fiber, quality carbohydrates. And of course, a dish of pulses and cereal provides a balance in essential amino acids for our diets. Despite all efforts, there lies a huge gap between research station and farmers' field yields. I would urge you all to look into this issue, where newly developed improved technologies should be scaled out and up with farmers, and the recommended packages are enhanced for a much higher uptake. Total production can be enhanced horizontally by bringing fallows land under pulse cultivation during fallow period. One of the fallow scenarios is the rice fallow in South Asia. And I understand that the government of South Asia have launched dedicated missions to bring pulses in rice fallow lands to break the rice monoculture, increasing cropping intensity and improving the livelihoods of the household and nutritional security across the region. ICARDA is an active partner, not just with India, but also in Bangladesh, Nepal, and government of Odisha, West Bengal, Madhya Pradesh, and then working with partners like IFAD, OCP, and others. In this context, and among the various dedicated programs to enhance pulse production, we are launching a global initiative called the Dry Arc, on which we hope that India will have a very strong role to play, with many of our partners contributing to the success and research to be undertaken under this initiative. It's basically led by ICARDA on rate-fed farming systems and can contribute for the transformation by scaling pulses technologies across many parts along the Dry Arc area, which goes all the way from Morocco up to China. So, in addition to what have been done, several research agendas warrant further development, including not a comprehensive list, but there's definitely greater work that is needed in the area of pests and disease arising from climate change, multiple disease resistance and appropriate integrated pest management packages to enhance the development of individual pulse crops, enhancing breeding efficiency, speed breeding. That's an essential part of the modernization of breeding programs under the one CGIAR, and of course using more advanced breeding techniques, including gene editing, and of course India has a lot to offer in this space. Pre-breeding approaches to winding the genetic base using genes from wild species and primitive land races, mechanization for seeds, from seeding to processing for pulses, and so on and so forth. I will not take too much time on these elements, but I want to emphasize in particular further work on geo-agro technologies that are to inform the decision-making and decision support systems. And I think India, again, is leading on this space, and we are trying to take experiences from India and share them in many other parts across the world, including in sub-Saharan Africa as well. So pulse crops were and will remain a crucial part of the dryland production systems of Africa and Asia by virtue of their importance in food, feed, and sustainable farming system. So research for development on pulses with low-cost technologies will be particularly benefiting smallholder farmers in marginal lands. Again, India has enormous experience that it can offer to many parts of the world on this space. ICARDA will continue to contribute under one CGIAR to these research programs, and we hope with our ability to come in a much more stronger and coordinated way to bring in stronger research support to our Indian partners and research, national research systems, state agencies, as well as many of the universities. We should be able to have a much stronger value proposition that we can bring to pulse research and then pulse production to help India become a world leader and then gradually achieve self-sustenance in this very important strategic product for India and for the world. I thank you for the opportunity to address you and wish you a very successful continuation of the scientific discussions today and tomorrow. Thank you very much.
Thank you very much for, sir, acceding to our request to address the gathering. And we are really very grateful that you accepted our request and addressed the people. Friends, just I would like to inform that Mr. Ali Sabah joined, DG ICARDA at a time when there was a crisis, in the Icarda, Syria. And he was the key person who, after taking the responsibility, the moved Icarda out of Syria and through a very decentralized approach, and he really stabilized. And one of the center, that legume platform that is opened, in India, Amla in Madhya Pradesh, which is again very adjacent to our center in Bhopal. We are working together, and in India, we feel when even it becomes a one CG centers, still Icarda will have a bigger role to play, in pulses. Sir, we are working together, and I assure that we will continue to work in the future also. Thank you very much.
Thank you very much. And just a small correction, I only continued the work started by my predecessor, Dr. Mahmoud Saleh, who's known to many of you, just to make sure that we operationalize the process for decentralization of Icarda, and also to scale up our collaboration with Indian institutions and Indian states government. Thank you very much for the opportunity.
Yeah, sure, sir. Thank you very much. Now it is over to Dr. Uday Kumar.
Yeah.
Dr. Uday Kumarji, to continue further.
Yes, sir. Thank you very much, Dr. Madan Pal, for an excellent presentation that you made, actually using diverse technologies of the approaches, screening approaches. You have given us a fairly good insight in terms of the response of the chickpea genotypes to different the temperatures. And it is quite impressive to hear the role of the calcium in the whole scenario. And I'm sure that you are going to bring in some more insight in terms of the basic aspects of how the calcium signal is bringing about enhanced tolerance to the high temperature tolerance is concerned. Thank you very much for an excellent presentation and for a comprehensive insight that you have provided in the genotypic response to high temperature, and your insight of how one should go about it and the predicted changes that are likely to occur with the 3-4 degrees changes. And your recommendation is quite valid in this direction. Basically, we have now approximately about 20-25 minutes time. And we have very renowned and the panelists here with us. They are going to bring in some discussion on the major topic about the abiotics response, which was skewed for today more on the high temperature. To some extent, the drought aspect is concerned. So we need to address and then get some idea from the experts like all of you and the panelists basically also on 3 major aspects with regards to the pulses is concerned. Of course, one basic question that still remains here is that is really the pulses that we talk about have relatively low yield potentials when compared to other cereals and etc. That is number one, which we need to give some debate and some insight and some interaction from you, number one. Number two, do we have, based on the experience of the number of genotype that has been screened at ICRISAT, both on the lentil as well as on chickpea, which has been very elegantly presented by Dr. Harsh Nair, do we have adequate genetic variability to identify temperature resilient genotypes to address the likely change in the high temperature to sustain the productivity aspect is concerned, and we require some input on this aspect. Number three, if you believe we don't have, is it necessary for us to introgress either by conventional breeding or by molecular breeding or by genetic engineering approaches to introgress some specific traits to enhance the temperature adaptation, which is likely to happen at least in some parts of the country, apart from other places? Is it necessary for us to launch programs in terms of introgressing some specific traits to improve the adaptation of our crops to high temperature, which is likely to come in? And if so, the fourth question that comes is, do we have the traits identified? Which are the traits which we should look for if we wanted to introgress, either to sustain the productivity of the high-yielding varieties, right, or to tailor certain genotypes of physiological breeding to enhance their adaptation to the high temperature, and in addition to the drought aspect is concerned. Do we have some sort of a leads in this direction? What type of the traits that has been crystallized so far from those who are working, both from your own work and also from the literature aspect about it? We'll try to, I would say one aspect is concerned, as very nicely as presented by Madan Pal, that the moment you increase the temperature, the crop growth duration coming down. We know about this aspect. It is a universal phenomena, which is now being looked into at the molecular level by a phenomena, call it as thermomorphogenesis, right? Not photomorphogenesis, but thermomorphogenesis. And we know the important role of a key molecule in terms of the P4 playing an important role in terms of bringing about acceleration of flowering and thus reducing the duration of the GS3 and the GS2 phases, which is likely to bring in significant reduction in the productivity, both by the number of sinks as well as the size of the sink as well is concerned. That's such information, and we know at a phenology level, there are certain traits which we need to look for, which itself has come out, and as rightly mentioned by Dr. Nair, in the sense, the pollen viability, stigmatic secretary emerging as the powerful, important responses with the plants and etc. Are they emanating as tools for us for screening? If so, do we have adequate amount of technologies to use these aspects to bring them together, or to screen and identify these subcomponents of the adaptation aspect is concerned? If you can spend the next 15-20 minutes on these 4 aspects, then it will be very nice, and we can provide certain leads for the discussion, and we can gain from your expertise on these aspects at this particular juncture. Now, with this small introduction on the presentation and need for look into these 4 or 5 aspects, now I would request our panelists, Dr. Venkateswarlu, you have any specific points to add on, and subsequently Dr. Chinnaswamy, Vishwanathan, and Dr. Rane at this particular juncture, please. Of course, Basu is there participating all through, and he will give his feels and opinion on these aspects.
Thank you,
Dr.
Dr.
Sir.
Yeah, thank you, Dr. Uday Kumar. Well, I will not be able to specifically, comment on the specific points you made, but given my background as a director CRIDA, I would like to generally add my own views on this whole aspect of abiotic stress management in pulses. And the first and foremost thing is that the abiotic stress management has 2 components. And one is the plant component, second is the management component. And in all these 3-4 lectures, of course, given their background that they're all being breeders, they did not touch a little bit, on the management aspect. I will just tell you one example. When I was a vice chancellor in Marathwada, we ran a campaign in 2016, 2016, I think the pulses prices skyrocketed, and we ran a campaign to increase the area under PGNP and to produce more pulses in the state. And the farmers responded, and then of course there was a good area expansion. And in that we introduced one single intervention which made such a dramatic difference that we showed them, that we demonstrated to the farmers that with all the water that you grow sugarcane, you can give one protective irrigation during flowering to PGNP in 6 acres. 1 acre of sugarcane, whatever water you save, not to stop sugarcane by going for drip, by shifting from flood irrigation to drip irrigation in sugarcane, whatever water you save, you can irrigate, you can just give one supplement irrigation to PGNP, and then you can double the production of pulses, which we have demonstrated. And that was a very good success. And then the state government has picked up that idea, and they are still running that idea, that to regulate the area under sugarcane, particularly in highly drought-prone areas, and then go for supplemental irrigation for PGNP, especially for PGNP. The second point I would like to make is that pulses, of course, they are already, they're, definitely relatively compared to cereals, they are already quite drought tolerant in the sense that, for example, if you take PGNP, even 30-35 days dry spell in the monsoon time, there is, of course, there is a retardant in growth, but, the crop still lives, it survives, and it picks up the moment there is a revival of rainfall. Of course, the problem is during reproductive stage, if there is a drought, dry spell and all, there is still a mechanism that there is a flower drop, and again there is a fresh flush of flowers, and such a resilient crop. It's an amazing crop actually for Indian conditions. As far as chickpea is concerned, thanks to ICRISAT work and also some of our IIPR's work, the heat tolerance work, I think, has already given us dividends. For example, chickpea, which was supposed to be a North Indian crop, is now grown large areas in South India, particularly in Andhra Pradesh, where I live. It is giving remarkable productivity in very, very mild winters. And the highest area now in chickpea, I think, in terms of production also, Andhra Pradesh contributes very large quantity. So therefore, chickpea, I think, as far as temperature tolerance is concerned, we have already given the results to the country in terms of the germplasm, in terms of the management practice, because the whole crop is mechanized in Andhra Pradesh. So therefore, it has completely replaced tobacco crop, because, and also many other crops in terms of economics, because of mechanization and because of heat tolerant genotypes, it is really suitable for peninsular India now. So these are two very good examples of, how our research has already helped in producing both PGNP and chickpea more. The remaining green gram, black gram, of course, there the drought and heat not so relevant because their duration is so low and hardly 60-70 days. But there the problem is, I think, heavy rainfall and also the diseases, pests, mosaic virus and all. So that's the issue there. Whereas as far as PGNP is concerned, as important as drought is the waterlogging. In central India, it is projected to have more intense rainstorms in future. And in Madhya Pradesh, we found the region furrow system of planting, broadbed furrow system of planting, it can save almost 60% losses during heavy rainfall. 2013 we have demonstrated, and 16 also. So I think these are two very good management practices where you can really maintain. And in eastern Uttar Pradesh, PGNP rice intercropping system, where the PGNP is grown on ridges and rice in the furrows. So that is an excellent system. Dr. Mangalray, earlier DG, has seen those trials of our Ecribda, and he really appreciated that. I think instead of toiling long way for breeding, we should really take advantage of these short-term, already available technologies which are already in our basket. So I think, and then final comment is that, we must actually try to, now with the remote sensing and all the tools available with us, we must try to actually divide and map those areas in the country where protective irrigation, just one protective irrigation can double the yields, and then try to, bring in some irrigation facility, if necessary, even diverting from other crops, providing some policy incentives, etc. And then the other aspect is where soil health improvement can boost the yields, So those areas also we have to actually map in terms of deficiencies and all that. So with these 2-3 interventions, Dr. Uday Kumar, I think we will be able to contribute to pulse production in a very significant manner, while the breeding, the marker assessment selection work, of course, is a long-term work, which still should go on. But this is the kind of balance I expect from this session. Thank you very much.
Thank you very much, Dr. Venkateshwar. He has given entirely the different side of the importance of the management, which has been missed in our discussion by all the those who has presented today, and especially your insight and then the information that has been generated to substantially improve the yields with one simple supplementary irrigation in the pulses suggest the resilience nature of the pulses, and at the same time, the high water productivity that one could achieve by diverting a small amount of water from the water guzzlers like rice and sugar cane in this particular direction. Of course, we have been concentrating very heavily on high temperature, which is the topic today, and also the drought. But at the same time, your insight and the experience has given us a point for us to probe on further how important is the inundation of water in this particular direction aspect. We'll come back again, sir, to you as the discussion goes on to get your wisdom and inputs to address the important aspects further. Now, may I request the other panelists, my friend, Dr. Chinnaswamy, Dr. Vishwanathan, please, if he's around.
Yes, sir. Good afternoon, sir. Thank you for the opportunity. Sorry, I could not join the session. I missed the first two lectures. Anyway, from the summarization by you and Dr. Venkatesh, I captured those points. I think it might have been discussed, and from the Madan Pal's presentation also it was evident that as we discuss for abiotic stress tolerance in any crop, the managed stress phenotyping for the component trait is very, very important for systematically improving the stress tolerance. As for example, when Dr. Madan Pal presented for the chickpea heat tolerance. So in our division for long, this heat tolerance experiment was going on. So we discussed maybe a few years back when we use a delayed sowing approach, it is not the high temperature stress that affects the yield to that extent. Rather, it is the reduction in the growth caused by the low temperature in chickpea is one of the very important cause. If you see the biomass accumulation by flowering time in the delayed sown chickpea crop, it is very, very less as compared to the normal sown chickpea crop. So if you have managed stress phenotyping facility, and if you phenotype for component trait, and the phase-wise phenotyping is critical to combine the tolerance mechanism together to enhance the stress tolerance level. And as rightly said by Dr. Venkatesh, the Kharif season pulse crops, they may suffer both from the waterlogging stress and to certain extent maybe by drought stress, but salinity and waterlogging may be an important stresses that we can target for the Kharif crop. Whereas the cool season legumes, both low temperature stress tolerance at early vegetative phase and the heat tolerance at reproductive stage will be critical. And when we do the component-wise trait analysis, it will be useful to map the QTLs for various trait, and we can introgress the QTL. When compared to the improvement in cereals, the QTL mapping has not been that much progressed in the pulses. So like if you see many of the pulse genome sequences are available, so a lot of genetic resources are there from chickpea, pigeon pea, lentil, mung bean, and faba bean, and common bean, cowpea. So various genome sequences are already available. So if we can combine the managed stress phenotyping with the genomics, we will be able to make progress quicker. And the duration of although the pulse crops are resilient, actually we need to vacate the field in certain fixed time. So we need to see if besides the resilience in terms of surviving and coming back to flowering again, or arresting the growth and waiting for the stress tolerance, if we can breed short duration pulses, especially the like crops such as PGNP, it will be very useful to enhance the productivity. And towards this, a lot of work is in progress. And the rapid generation advancement method, as mentioned by the Director General ICADA, is very key. So currently for chickpea, to some extent, it has been standardized. But I think for the crops like PGNP, that will be very important for the long duration crop, where we can do rapid generation advancement, as well as the synchronization of flowering between the wild relatives and the cultivated ones, so that we can do pre-breeding to transfer the trait from the wild species into the cultivated species. And to target the rice fallow area is one of the major goal of the government since long, but still we have not achieved that target. So is there is any possibility to capture the rain fallow, the rice fallow, as well as the wheat fallow? For wheat fallow, definitely heat tolerance will be very important. And finally, the gene editing technologies will be definitely useful. So we can, as Dr. Venkateshwar told, the pulses to some extent they are stress tolerant. So we can see what are the trait or genes that we need to take with the gene editing technology, or if we can remove the toxins such as in Lathyrus, we can produce pulse crop with enhanced nutrient quality. So that is in brief, sir. Thank you very much for the opportunity.
Thank you very much, Dr. Vishwanath. You have covered a wide areas. And of course, the message is very clear that we need to go for improvement, genetic improvement of the component traits for further improving it, and then adapt. The QTL technologies were already done in the other pulses, other crops, which needs to be adapted in this particular direction as well. And in addition, he also has given some insight about the need for paddy fallows as a management options, and then improving the productivity. But I would like to ask you one quick question for the benefit of everyone today. Apart from the adaptation aspect about it, do we really think that we need to put stress on improving the yield potential, intrinsic yield potential of our pulses? Is that an important, mainly because of the fact they have been adapting it fairly to the higher temperature and the drought, as Dr. Venkateshwar was mentioning, their recovery growth rates are much, much higher because of the semi-determinate aspect and etc. But is the yield potential is a limitation, and how much stress that we should put in this aspect? A quick question quick answer from Dr. Vishwanathan, from response from Dr. Vishwanathan, and any other please on this aspect. Dr. Vishwanathan, you have to unmute yourself.
अनम्यूट करने के लिए
thank you, sir. Yes, it is a very important aspect, sir. Definitely, we need to increase the productivity of pulses. Although the stress tolerance is important to achieve the potential yield, even the potential yield is not very high. So definitely, we need to improve the potential yield of pulses. So although if it may be very difficult in very short duration pulses such as summer pulses, summer bean or other thing, we can improve the productivity of PGNP and chickpea, which are the major pulses. Definitely it is needed, sir.
Very good, very good. It's very nice input. And actually, the Dr. Sheshasai has touched quite a bit in terms of the options and the approaches that is needed for the yield potential. You have any quick answer, Dr. Sheshasai, on this? Are you around, Shesha?
Yeah, he's there.
Yes, sir.
Can you give your opinion quickly? Very focused reply on this, so that we'll go ahead further with the next.
The one word answer is yes, definitely. I mean, as Vishwa said, and in the literature also, it is very amply clear that the yield potential itself under normal conditions and optimum conditions itself is low for pulses. So any attempt that is made towards improving that can definitely boost productivity other than the all these abiotic stress and biotic stress tolerance.
Is it associated with the sink constraints, or we have anything connected with the growth rates itself?
From the literature that I read and understand, I think both of them seems to be a problem, sir, in the sense the source is not that very high, as we can evidence from the number of experiments that people have done with the elevated CO2 and giving more light and other kind of artificial ways of doing things. There is an improvement in yield. And the major thing seems to be the reproductive size or the sink size in terms of the flower drop, fruit drop, abortion, which Dr. Nair was talking about. Under high temperatures, the pollen become bad, and under any stress, this is what happens. And even under normal conditions, there seems to be some kind of a the sibling rivalry associated abortion of the ovules. Some basic efforts in terms of understanding and then going ahead for the breeding to improve that will have a definitely a better influence on this.
Thank you, Dr. So we'll come back to you with many more clarifications and inputs. Now I'll request the third panelist, Dr. Rane, who has been really contributing immensely in the Abiotic Stress Institute in the area of several crops, including the pulses. Dr. Rane, please, your opinions.
Rane, unmute yourself, Rane.
Rane, yeah.
You are muted. Unmute, please.
Yeah, thank you, Madan Palji. I forgot that one. So just I was telling that there are some of the key points that we have to take into consideration when we talk about pulses. They do not only like meet the food requirement of hunger of a human being, but also the soil and soil microbes and all. They are adding to the ecosystem services that cannot be ignored. But a very important question asked by Dr. Uday Kumar is about yield potential. Yes, yield potential is less, but that again is determined by the environmental conditions. When we talk about yield potential, it should be irrespective of environmental conditions. But what I feel, being a kind of semi-perennial type of plants, they are capable of producing more biomass. The limitation must be converting that biomass into the yield. That aspect has to be taken into consideration. One more point that we have to take into consideration is current focus all the time about is genotype into environment interaction that we are talking about. And all the time, all these pulses where the crop of marginal land where resources are poor. But if the mandate is to increase the pulse yield with giving more resources, we can increase the yield of pulses also. That is possible. But one thing that we have to remember that G into E into M, that's the management part that we have to take into consideration to address the issue which pointed out by Dr. Venkateswarlu. That is very important. When we talk about water logging, then there is a good management practice with raised bed there, even for the pigeon pea. Are we really looking for the traits that can contribute to get better yield under that type of agronomic condition? That is really very important. Then what I really feel is the success strategy should be like we are focusing on visible traits of shoot and also some to extent root and all that we are talking about. There are many invisible traits. There are protective mechanisms and all. We have to look into the surrogate traits to look into that aspect. For example, osmotic adjustment and then antioxidants, and we are talking about which is not really feasible to screen large number of genotypes for these traits. Are there any indirect traits, surrogate traits that we have to look into? Then when we talk about mechanisms, we have explored the escape mechanism, the short duration varieties. So we are happy with that one. But someone just pointed out the diurnal escape that we have not studied. How these pulses, they are capable of using that mechanism of diurnal escape as demonstrated in what we call rice by Jagdish Krishna and all. Then we talk about all avoidance mechanism by sufficient tissue water level. So many of the studies, if you look into the look into the abiotic stresses, many of the consequences of abiotic stresses have been reported. All the consequences are not adaptive, considered as adaptive traits. How to prevent that adverse consequence, and what are the mechanism existing there, and what are the trait they can explain that. We have to focus on that one. So another point is like what really we are searching for, key or the treasure. See, many times we revolve around only the yield, but there are many other traits. For example, the pollen viability or pollen tube growth and all so many other things. They are really responsible ultimately to convert partitioning of biomass into the yield. They are the key points there. So are we really able to screen a large number of genotypes? Then other point is that already, like if you look into the one of the review by Dr. Basu's team, it is a Jha et al. In 2014. For every abiotic stress, at least there are 25 genotypes as registered as a genetic stock. Are we really using them? Have we looked into all the traits that we are talking about? That is very important. Then the last couple of points is that, last couple of points is that like we are many times we compare the highly sensitive and the tolerant one. So what we have to do is we have to take the popular variety as a reference rather than the sensitive variety. We have to shift our approach. So what is the limiting factor in the highly popular variety, that we have to look into, whether pollen germination, pollen tube growth and all, they are really a problem there. If so, how much fold better value, like how much fold higher value we are getting in the genetic stock, that we have to look into. And yes, so that's what I wanted to tell, and I wanted to add some of the references how we can go into that in the second round if I get. Thank you, sir. Thank you very much.
Thank you very much. I'm not very sure how much time that we have, but a very, very comprehensive input given by Dr. Rane, while emphasizing actually the mechanisms, while emphasizing the need for us to identify the traits for adaptation and the yield, right? I also agree with him that we must be in a position to initially, as a first step, to identify certain surrogates so that we'll make the next step in this particular direction of trait-based associated aspect, and also get going to have a clear understanding about the relevance of the duration as an escape than for diurnal escape mechanisms are most relevant still to completely exploit the growing duration is an important, and we need to get in that direction. His input apart from the management are very, very useful for those working in this particular area. I will not be in a position to elaborate much, although I have a few points, Dr. Rane, which we'll discuss later because of the time factor. And now I should request, thank you very much, Dr. Rane, for your excellent inputs and a comprehensive insight that you have provided, how to go about it and how to address certain of the important issues. I need to ask his comments from the fourth panelist, Dr. Basu. And
Yes, sir.
I would like to request you to be a little more brief so that the time is the one that is given is we are just passing through. Please, Dr. Basu, to hear you.
Am I audible, sir? Am I audible?
Yes, Dr. Basu. Yes, Dr. Basu.
Sir, as a panelist, I have little to contribute or some given because already most of the things have been covered. But I am at the same time convener of this session also. So at the outset, I express my deep gratitude to all the leading scientists of the nations, Professor Uday Kumar, Professor Venkat Sarulu, Chinu Swami Vishwanathan, and Jagdish Rane. At the same time, speakers who is very expert and who covered the physiological, genetical mechanism, signaling effect, and almost all advancements, what is really happening in the research area of abiotic stress has been covered by Professor Sesai, then Dr. Harsh Nair, and then the Madan Pal. So as you have heard that they have given the wide coverage of these things. So only I have some points and just to say debatable questions like that. So as Dr. Harsh Nair has shown in his slide, that is the contrasting heat genotype, heat tolerance genotype, sensitivity is different, and he could identify many of the genotypes which is heat tolerant and which is also at the same time heat sensitive. I had the opportunity to work with the CRISAT in heat tolerance, also Harsh Nair. I have some fortunate enough to other speakers and panelists to whom we work together. But you see that when question comes about the drought tolerance, you see that as Dr. Venkateswarlu says that he already pulses as a whole is a drought tolerant species. And we have to explore that key, what is the further scope to improve the drought tolerance. Say, for example, the drought tolerance in chickpea. We had a long collaboration with the ACIR Australia, where both of the countries were worked together to identify the potential traits which impart tolerance to drought tolerance, drought stress. But you see that none of the literature, although based on some specific traits about root and all these things, osmotic adjustment, people had declared that this particular donor has been identified as a drought tolerant. But very little literature or almost no information about information is available about the drought sensitivity in chickpea, because no reports I have seen that people have already declared that this is a peculiarly as compared to the heat tolerance, there is gradations, but drought tolerance, that kind of the contrasting lines we don't have. So there is a question comes whether the chickpea is already reached to the threshold level of drought tolerance, and is there any knowledge gap? We could not pick up all these things, and what is the further scope to improve drought tolerance in chickpea? So because you know that what we are working together with the plant breeder, they want something else from us. They want rapid, fast screening method so that they could identify non-destructive way to identify the genotype which is drought tolerant. But you know that the physiologically lot of sort of things, physiological mechanism, and then traits we are having, we have got the precision phenotyping with us. In spite of everything we are having, the progress in the drought tolerance, in particularly chickpea, and I am the active worker in this chickpea, or no significant breakthrough comes in the drought tolerance in chickpea. So we have to revisit these things, say whether we should focus on some other pulses crop which have still scope to enhance the drought tolerance. So this is the my observation is that, and there is no straightforward answer. Is if breeders are asking that you come out with a potential traits, on that basis screen can be made. There is no such type of answer. It is a complex traits. And talking about the droughts, we can say that the two type of the drought we are facing. One, the terminal drought, and another is the in between some intermittent drought, or initially there is a drought. So because you see the terminal drought, what Dr. Rani is saying that by management practices we need to improve along with the physiological tools or some kind of donor which is improved drought tolerance. So what is the mechanism of improving the drought tolerance for the terminal droughts particularly? If we are able to build up biomass at initially, there is to large extent we can control about the drought yield reductions. And another thing is that if initial drought is very heavy, then even chickpea, so much resistant crop, so far drought tolerance is concerned, we cannot address. These issues are this, management practice is very, very important. And the way the physiological advancement at molecular level is going on, I appreciate. I am thankful to Professor Seshai, Dr. Harsh Nair, and Dr. Madan Pal for the wonderful contribution made in various aspects. Thank you very much.
So thank you very much, Dr. Basu, for giving a very comprehensively the need, both for the management as well as the identifying the traits and the present scenario with regards to the drought and lack of considerable number of the genotypes for the sensitive types and so on and so forth. But we'll come back to the test. I just have a quick question to Dr. Harsh Nair, whether the information that he has provided in terms of the pollen viability and the stigma receptibility to the high temperature, do you have any specific insight with regards to waterlogging and also the drought on these two traits, Dr. Nair?
So not on waterlogging, but on drought stress, we have done some work on soybean, not on chickpea, exactly, on these traits. So, but we are mainly focusing on heat stress. One of my students has worked on combined drought and heat stress, but on seed filling aspects. So we haven't generated any information on floral function yet on drought and combination of drought and heat as well as flood. But we can definitely go for drought and heat combination on floral function. But it's very difficult to work on these two combinations. Very difficult to impose these stresses in the field because there are many complexities involved in providing these two stresses together in the field situation. Now we have walking growth chambers, which are quite good, but still I won't say that that will be a realistic situation in comparison to the field. But
what about low temperature, Dr. Nair?
So low temperature, we had a project, you were a panel member at that time. We have done excellent study now on the floral biology. The final report will be submitted in detail within few days, which will be coming to you. We have published recently in Plant Cell and Environment also on the pollen biology under cold stress situation, where different stages have been quantified. We will send you that paper. Specific stages have been captured, which are showing sensitivity. And that is one of our excellent contribution for the cold stress. And we would like to extend that in heat stress situation also this year.
Because the cold stress is the one that very emphatically raised by Dr. Madan Paul, which is also coming in the way, especially under certain growth stages aspect is. My last question to Dr. Sesha, do you think that we have adequate variability in one of the important plant traits, that is the water productivity, which we often call it as the water use efficiency in pulses? What is actually the literature says, and what is your opinion whether we need to look into this particular aspect? Do we really have requisite amount of the variability, and also how they compare with some other crops like the cereals on this? Dr. Sheshasai, please.
Yes, sir. That's again another important question to answer. The literature, there is quite a bit of differences. We did make measurements on the germplasms of groundnut and chickpea when we had that program with the ICRISAT. And when we use the stable isotope ratios, there is about 3-4-fold variations that we find in water use efficiency, which can be really used for breeding. And the kind of groundnut experiment that has been done, reported in the literature as well, there is a lot of variation in water use efficiency, though most of them have been using the surrogates like the stable isotopes. But except for a few things that we did with about 10 or 12 varieties of moonbeam, there is some good variation, but then the actual measurements of water use efficiency has not been done in that way as much as has been done for other species.
Thank you.
Okay, somebody is talking. So I have a feeling that there is a lot of variability that exists in some of the important drought adaptive traits, especially. I mean, I thought that instead of going into the details about when Dr. Basu says drought is a complex trait, like Blum was saying once, if you can continuously say it is a complex trait, it will only distract people from getting into that. We can say it's a difficult trait, but we will simplify it from the physiology point of view. Looking at water use efficiency and the root traits and the cellular level tolerance for the intrinsic, the acquired tolerance traits, if you can combine them, I'm sure there can be a significant improvement in the growth. But then you look at the reproductive biology, there can be a lot of improvement in total productivity in terms of.
Thank you. Thank you, Shesh. Actually, I recall an interesting program with Australian group in terms of combining the traits in terms of the transpiration efficiency and the water mining aspect about it. And several institutions are involved, and quite a good material has been generated in the groundnut. And that's the one that I was trying, whether anything has been done in CISR and also in the other pulses aspects.
After that, we did try getting that into the ACIAR programs and the Indo-Australia grants, but somehow or the other it did not go through for whatever reasons, but doesn't matter, sir. But what I'm saying is that when we combine the trade, that's what I would say, when you combine water use efficiency with water use, and that was the kind of concept that we had taken forward in that particular program in the 1990s, late 1990s. So
thank you, Sesh. Thank you. Yeah, I got it. I got your message. Yeah, you conveyed very nicely on this. Thank you very much, Dr. Sesh.
I think there are some questions from the audience also, sir.
Yes, sir.
I do not know, Dr. Basu, can you apprise us how much time that we have, whether we need to conclude the whole session? If so,
Sir, actually, immediately after this session, we are going to start the poster session of the same the topic, abiotic stress management.
Okay.
So just we want to close these sessions with your permission, Robert.
Yes.
And then,
Yes.
sir,
Before you close it, let me thank once again all the panelists for their excellent input, and also the speakers who has given a fantastic insight and a good discussion that we have. Now I request this convener to complete this session.
Yes, sir.
So that, please,
yes,
please go ahead, Dr. Basu.
The president ISPRD, our society, is proposing a vote of thanks. Thank you, President.
Good afternoon to all of you. Although Dr. Basu has briefly thanked to all, but as customary, on behalf of ISPRD Society, which is organizing this webinar, I am going to extend a vote of thanks to all speakers and members. So first of all, I am very much thankful to Professor Uday Kumar, who is very renowned plant physiologist and has worked in different capacities at US Bengaluru. So, sir, I am grateful to you for accepting our request to moderate this session, and we have conducted this session very nicely in a proper manner and within the given time. I am also very much thankful to our Dr. Sisai from US Bengaluru for giving exhaustive talk on improving productivity of pulses under water limiting conditions. Then I am very much thankful to Dr. Harsh Nayyar from Punjab University, Chandigarh, who has delivered a thought-provoking talk on impact of high temperature on food, legumes, and approaches to improve heat tolerance. I am very much thankful to Dr. Madan Pal, very good physiologist from IARI New Delhi. He has given talk on phenotyping of chickpea for drought and high temperature state tolerance. And I am very much thankful to all the learned panelists, including Dr. B. Venkateswarlu, former Vice Chancellor of VNMKV Parvani, Dr. Chundu Swami Vishwanathan from IIR New Delhi, Dr. Jagdish Rane from NISM Baramati, and our senior colleague, Dr. P.S. Basu at IIPR Kanpur. So again, I am thankful to all of you, and also I am thankful to all the listeners who have listened the talks very patiently. Thank you again. Thank you all.
Thank you very much, sir. We leave the...
Thank you, sir. Dr. I.P. Singh, President ISPRD and Co-Chairman Pulse Webcon 2021. Within a span of 5 minutes, we will be initiating the next poster session for the theme Biotic Stress and its Management. We will have a short break for 5 minutes to set up the infrastructure, and I welcome you back within 5 minutes for the poster session. Thank you.
क्या हो रहा है? अभी भी नहीं जा रहा। ये हो रहा है, ये दोनों ही एक जैसे हैं। ये फुल अच्छा अब ये है ना? तो ठीक है।
Start. Good afternoon. This is Akram from IIPR Kanpur. I'm going to display the poster of the session 3, Biotic Stress and its Management. The panel of judges are Dr. D.R. Saxena, Dr. Bansa Singh, Dr. Mamta Sharma, Dr. Poonam Sharma, and Dr. Kodandaram. There are total of 55 posters. I will display one by one. And then for each poster, I will give about 10 to 15 seconds. The judges are requested to ask the question, and the poster presenters, means the posters who's having the poster, they should be ready to reply the question asked by the judges. So all the participants who have submitted the posters in this particular session are requested to be attentive. Whenever the judges ask the question, they have to reply. I will just read the title and I will tell the affiliations of that particular research work done where. So this is my job only. So I will display the these posters one by one. So the first poster is Ankit. Ankit, it's not moving. Okay, this one. ये ऊपर कर हटाओ. Okay. So poster number S4-2, biointensive management of whitefly and YMD in mung bean in Udaipur of southern Rajasthan, India. And the first author of this poster is D.K. Jain from VB Krishi Vigyan Kendra, Badgaon, Udaipur. So if there is no question, then I have to move the next poster. The poster ID is S4-3. Combining bruchid and mung bean yellow mosaic virus resistance in green gram genotype to enhance productivity. From Prasanta Kumar Majhi, Department of Genetics and Plant Breeding, College of Agriculture, Dharwad.
Yeah, I have a question here. This is Mamta Sharma.
Yeah, ma'am.
I'm online. Dr. Suma is online from Dharwad. You can go ahead, ma'am. Yeah.
Yeah. So just wanted to know that whether these lines were how many times the confirmation was done for checking the resistance for mung bean yellow mosaic virus.
Yeah. It was done. It was done under confined experiments with the lab-raised white flies. We have only checked for the vector and not for the virus.
No, but did you repeat, confirmation once or twice?
Actually, this was the same material was used earlier in the studies. This was the third successive year that we used. And since these are the recombined, these are the products of recombination and mutation, ma'am, we found combined resistance to both MYMV and bruchid, ma'am.
All right. Okay. Thank you.
This is a consistent performance, ma'am.
Yeah. Thanks.
It was a part of my students' work.
Okay.
So shall I move the next judges?
Yes.
Okay. So the next poster ID is S4-4. Efficacy of novel fungicides and neem oil against chickpea rust, Euromyces ciceris arenati, from PR Sabale, Regional Center of Indian Institute of Pulses Research, Kanpur.
So I have one question again here, Mamta Sharma. So this was done under natural conditions or artificial conditions?
Sir, madam, it is done under field conditions.
So under field conditions, so you have not inoculated, but under naturally infested plants.
But it is very difficult in artificial conditions because this fungus is this one obligate.
Okay.
Yep. Okay, thank you.
It was done only one season.
Yes, madam. Yes, madam.
Okay. Thank you.
Okay, thank you.
Hello.
So the next poster ID. Yeah, you have, sir, any question?
I have some clarification here from him. Which fungicide do you find better in controlling the disease here? Hello.
Sabale, please reply.
Hello. Sir.
Which fungicide was effective against the rust here? And how many years you have? Only 1 year data?
Actually, I have done this year also, sir.
Hmm.
But it is I have to compile that one also. But native one fungicide is there, tabuconazole plus trifloxyflobin. one fungicide is there that is very effective for rust management, and also score, that is difenconazole.
What about the economics here?
This year also I observed same fungicides only, very effective for rust.
So you have you calculated the economics of it?
I have to calculate, sir.
Okay.
I will calculate, sir.
Okay.
Yes. Okay. Thank you, sir. Okay.
So the next poster ID is S4-5, induced resistance in grain legumes against insect herbivores through exogenous application of signaling molecules by Shivani Kokar from Department of Entomology, Punjab Agricultural University, Ludhiana. स्क्रीन ऊपर आती है, ये नीचे आ जाए, जो ये टाइटल पढ़ने में प्रॉब्लम होती है, वो जो ऊपर दिखाता है वो ऊपर So if there is no question, then I have to move the next slide.
Okay.
Excuse me, sir.
Yeah.
Sir, what is about session third poster presentation? You have started directly as for yes, so I am asking, sir.
Session third.
Huh?
So these
session third only. Fourth. You have started yes for, sorry. So I am asking, sir.
No, actually we have already given the number and then there is some shift in the program. So already we have communicated to the participants that same number. Though the session is 3, but the ID we have given, it is S4-5 like that.
Okay, thank you, sir. Thank you.
So the next ID is S4-6, insect resistance transgenic chickpea harboring insecticidal WIP3A for resistance to gram pod borer by Prateek Singh and team from Indian Institute of Pulses Research, Kanpur. So the next poster is S4-8, occurrence of green gram weevil, Pechyticus mungonis on black gram in northwestern Himalayas and its control by S.D. Sharma from CSK HPKV Hill Agricultural Research and Extension Center, Bajaura.
Hello, here I have one question.
Yeah, please.
You have said that it is economical. In what way you can say it is economical?
S.P. Sharma is not connected, I think.
Okay.
I think.
Okay, you can move next.
So the next poster is S4-9, in vitro evaluation of Fusarium oxysporum udum isolates causing wilt of pigeon pea by H.C. Lal and team from Department of Plant Pathology, Faculty of Agriculture, BAU, Ranchi.
Yeah, so I have a question here. First thing is that in title, it's not mentioned against what we are evaluating, in vitro evaluation of what, like fungicides. And then I would like to know what is the application of that? How are we applying this?
Actually, in lab it was tested, fungicides, then soil amendments, and then biocontrol agents were evaluated in lab.
Okay. So how do you see its application in the field?
In application, it was also it was used as seed treatment, and it was carried out in another separate trial. It is not mentioned here.
Oh, I have got one question.
Yeah.
You said you tested soil amendments in lab.
Yeah.
How you have tested soil amendment in lab against
Actually, aqueous extract were first prepared. In crude extract were prepared 2:1 ratio, 1:2 ratio. Then it was evaluated. The procedures are mentioned, but through the limitation of the space, I could not mention all this.
Okay. Next.
So shall I move the next?
Yes.
Yes. Yeah. So the next poster, ID S4-10, management of chickpea will complex. Post-plant resistance, fungicides, organic amendments, and biocontrol agents by Savita Eka and S. Silal from Department of Plant Pathology, Faculty of Agriculture, BIU Ranchi.
I have got one question here. Bansa Singh is here. Anybody listening?
Yes. Please.
It has been mentioned wilt complex. Can you specify pathogens included in this complex?
Savita, you are listening?
I think not connected. Okay.
I think Lal, S.C. Lal is here.
Yeah, he can also answer.
Okay. So shall I move the next?
Yes. It's not connected, you move.
Okay. So the next poster is ID is S4-11. Molecular characterization and genetic diversity of Erysiphe.
More than one. No, bacteria भी पहुंच जाए थे पर कौन
Erysiphe by causing powdery mildew of pesi.
हाँ complete.
सामने आ जाता है.
So I have one question here. Was there any similarity in the molecular characterization and the pathological characterization if it has been done? Or it was only molecular?
So anybody from the poster presenter side? Parthisarathai. Anybody from team? Reply, please.
I think I saw Parthisarathi, Dr. Parthisar in the chat. So maybe if he can unmute and reply.
Yeah, please unmute.
Yeah. Otherwise, you can go ahead.
So the next poster ID is S4-12. Morphological diversity and molecular characterization of M. phaseolina causing dry root rot of pigeon pea from India by Monica Mishra and team from Division of Crop Protection, IIPR Kanpur.
Yeah, I have a question here that how was the pathogenicity proved for Macrophomina fezulina in pigeon pea?
Good evening, ma'am. Yes, ma'am. Hello?
Yes,
yeah,
we can hear you.
Ma'am, sorry, what's question?
My question is that did you prove the pathogenicity of Macrophomina and,
Yes.
and what was the methodology followed, the technique?
Ma'am, by following in a coach postulate, I just proved that pathogenicity of Macrophomina.
No, what was the technique used? Where did you inoculate? On the roots, on the stem, or where?
Ma'am, on the by inoculating in soil or by inoculating in a root? By soil, ma'am.
By soil. And in how many days you observe the symptoms?
Ma'am, 15 to 20 days after inoculating.
All right. Okay. Thank you.
Thank you, ma'am.
So the next poster ID is S4-13. It is not much visible. Potential use of some traditional plants extracts against Fusarium wilt disease of chickpea from Bhardwaj.
Yes, sir. It's mine, sir.
Bharadwaji, what can be the conclusion here? We cannot see it.
Sorry, sir. But main thing is, sir, instead of using, my work is basically on the, instead of using synthetic chemical, plant sex is a good substitute. This is the lab condition studied, and we have screened in this study 50 plant extracts. So labs against this particular pathogen. So Lawsonia seed extracts of Lawsonia enormis mehndi have shown the marvelous inhibitory effect against this pathogen, followed by this Acacia catechu, bark extracts of Acacia catechu, and rhizome extracts of curcuma. This is healthy. So my work is on this, sir, on this slide. Because synthetic chemicals are totally previous in dangers for environment as well as human being. So this is my work, sir.
What concentration you have used?
Sorry, sir?
What concentration?
15%, sir. Weight by volume 15% by food poisoning method. And I have seen the minimum inhibitory effect by hanging drop technique also, where 0.25 to 3% extracts was prepared to see the effect. So more or less vary 0.5 to 3% up to. So in here 3% extracts are more effective. So this is.
What can be the applied aspect?
Sir, this is a very, because plant extracts are easily available, and for environment, and synthetic chemicals are very dangerous for human being as well as the environment.
But how to apply? How to apply?
By preparing the extracts and spraying in the field, as usual, as we use the synthetic chemical.
So
we have already started field experiments are here.
So do you think that foliar application can control the Fusarium wilt?
Sure. That's why these are our results in lab condition, and we are now trying to see the effect in field condition also. So up to 3 years we will see. But this is the because plant extract possesses various secondary metabolites, which shows the inhibitory effect. Various literature show. So our studies also in lab conditions show that these extracts are very effective against this pathogen. So instead of using Regor, diphthane, and other chemicals, so these extracts can be applied. These are organic, cover organic aspects also.
I have that means these chemicals have got systemic effect. They are systemic if they enter the plant system and go to roots, isn't it?
Yes.
Hello, Dr.
Hello.
Bharat, can you listen me?
Yes, ma'am.
I have one question. How you have extracted these plant extract? Is the methodology is very easy? And are these
Yeah,
plants?
madam, we can even a layman or farmer can prepare the extracts by taking 15 gram in boiling in 100 ml water and prepare the extracts easily.
But we are the
Then he can apply in the field also.
These extracts also.
Because
We are the scientists, Dr. Bhardwaj. We should know the constituents of these extracts and which constitute is for inhibiting these organisms. I should
very
that part should be.
hello. Ji. Sorry. Someone called me. That's disturbance. Yeah, sorry. Yes, ma'am.
My question is that farmers, they are just extracting these material, but which is the constituent which is inhibiting these fusarium or any fungi of their growth.
So sure. Actually, madam, my works are covered two aspects. One for farmer, other for industry. When we know that this is the plant and it shows the effect against this particular pathogen or other pathogen, then we can assess to see their activity to find out the respective component which shows the inhibitory effect. Then further study can also be elaborated. Why we think about, because earlier in ancient time, I think there is no synthetic use synthetic, only organic or these crude product of plants are utilized at the time also. But
shall I move, judges? Please, we don't give much time only for one poster because we are having 55 posters.
Dr. Akram, otherwise a lot of posters are there.
Okay. So the next poster ID is
okay,
-14.
Thank you.
QTL related studies for Ascochyta. ये जो है बोला मैं, अरे अंकित इसको करो या तो वहां कुछ ना करो. Ascochyta blight disease in chickpea by Sangeeta Soy from IRI. या तो इसको नीचे करो, कुछ हमारी स्क्रीन जो है दिखने में, और या तो ये छोटा हो गया.
Maybe I have a question here. So meanwhile, I was just thinking, do we have resistance available for Ascochyta blight, and what is the resistant parent used in this study?
Yes, ma'am. The resistant parent used here is GL12021. And the susceptible one are PPG7 and Vijay as a Vijay is also taken as a susceptible and PPG7, and the resistant is GL12021.
What is the disease score level for this GL12021?
Score level, ma'am, score level is not clear to me. I can't tell you the score level. I'm sorry, I can't tell you the score level.
Okay, and the screening was done under tray condition. That's what I can see.
Yes, ma'am, tray condition. It was MABC approach, which has been followed. And BC3 F2 generation is in the field. The lines are being further advanced to successive generation for identification of superior resistant lines.
Okay, yeah, thank you.
Thank you, madam.
Next.
So the next poster ID is S4-17, screening on black gram genotypes for resistance against storage pest brookage by S. Ragul from National Pulses Research Center, TNAU, Mumbai.
I think this poster should have gone for post-harvest management, isn't it?
Of course, it's a biotic related work.
But under post-harvest management, it is stored grain pest.
It's fitting
That's
here.
what I think. Anyway, okay.
But brookid is a storage pest. We can use it as a biotic agent that reduce the yield loss now.
Oh, okay.
So that.
The next poster ID S4-18, seasonal incidence and extent of damage due to major insect pest of pigeon pea by DS Ahlawat from CCS HAU Regional Research Center, Rohtak. So the next one, S4-19, selective isolation of Bacillus thuringiensis from rhizospheric soils of Deoria, Uttar Pradesh, and its characterization by Sujayanand GK from IIPR Kanpur.
Hello.
Yeah.
Sir, myself Sujayanand, sir. Good evening, everybody, sir.
If there is no question, then I have to move the next poster. S4-21, studies on systemic induced resistance in chickpea against biotic stresses using novel chitin amended formulation of Trichoderma asperellum and Pseudomonas fluorescens from Pratik Jesani, Department of Plant Pathology, Anand Agricultural University, Anand, Gujarat.
The font is again.
So it's very poor poster, we're not able to read. So the next one is S4-22, characterization of transgenic pigeon pea, plants for presence and expression of CRY1AB gene by Manjinder Singh from Punjab Agricultural University, Ludhiana.
Good evening, everyone. Yes.
Hello.
Hello.
Yeah.
I have a question for you.
Yes, sir.
Did you do any bioassay for these lines?
Yes, sir.
Bioassay. Have you conducted any bio?
Hanji, we did bioassay on flowers and pods, sir.
Okay. And which method you followed?
Sir, for bioassay, sir,
No, which method? Is it diet method or
in vitro
feeding
method was followed, sir. In vitro bioassay method was followed.
No,
using
you have taken pod and given the for feeding, or what you have done?
so we took the pods and flowers and gave them for feeding to second instar Maruca vitrata larvae.
Okay, second mixture you have taken. Okay.
Yes, sir.
Okay, thank you.
Yes, sir. Thank you, sir. Thank you.
Okay.
So the next poster ID S4-25, comparative toxicity of insecticide formulations against tobacco caterpillar, Spodoptera luteura, infesting legumes by Rajesh Kumar from Chaudhary Charan Singh University, Meerut.
I have a question here.
Yeah.
Hello, sir. Kumar. How is the best for this Spodoptera? which insecticide was found better for the Spodoptera?
Sir, actually, imamectin benzoate was the
Hello?
most efficient we found. So we found imamectin benzoate as most efficient insecticide, followed by chlorantraniliprole.
What is the field dose, or you have standardized the dose in the bioassay?
Sir, actually, we have standardized 6 different doses. Then for each insecticide, then we have conducted a assay. So for every insecticide, we have taken 5 or 6 different doses. Then this was the we found that imamectin benzoate was the best.
And what is the method you have followed? Bioassay
Actually,
method?
we have followed artificial diet method.
Okay. Artificial diet method you have
Artificial
followed.
diet
Okay.
method we followed. Yes, sir. Yes.
Okay. Okay. Thank you.
So the next poster ID is S4-26, efficacy of insecticide against blister beetle in pigeon pea at Nagaland by Tinatoli Sima from ECRIP on pigeon pea, School of Agriculture Sciences and Rural Development, Nagaland.
Hello. Hello.
Sir.
Whether the person is here or not. How much
Seema,
incidence is there, blister beetle?
From poster side, the Seema or anybody from Kavi Sumi or MB Sharma? I think, sir, nobody is there.
Okay, next we can move next.
So the next poster ID is S4-27, screening of pigeon pea genotypes against pod borer and blister beetle in Nagaland. I think from the same group again. So the next poster ID is S4-29, population dynamics of pod borer, Helicoverpa armigera, Itella Zincrinella and Apis crassivora in relation to abiotic factors by DS Ahlawat from Rohtak.
I have a question for the presenter.
Sir, please.
But find out whether somebody is connected.
Yeah, Ahlawat or anybody. I think nobody is there.
Yes, sir.
Yeah, he's here.
Can you tell me what is the season you have taken population dynamics? Which season?
Yes, sir. Repeat, sir.
What is the season you have taken? Season, cropping season, whether it is a Kharif or Rabi season?
Rabi season, sir. The crop was sown in the month of November, 15th November 2017, and observation started from first week of January till harvesting of the crop.
Tea or what was any other crop? With the crop?
Lentil, sir, variety grima.
Okay, lentil. Okay.
Yes, sir, variety grima.
So the next poster ID is S4-30, identification of agromyzid flies on major food legumes of India. From Prabhu Ganigar from Department of Entomology, Bangalore.
Good evening, sir. I am here, sir, with you.
Not mentioned the conclusion. What is the conclusion here?
Sir, here in food legumes, no research, particularly on taxonomic research, sir. Here we have made a first attempt, particularly on food legumes, wherever they are growing. These all specimens we have collected, we have identified up to species level, sir. Based on the morphological characters, particularly Spencer, he worked in 1961. Afterwards, Spencer visited India in 1986. That time, afterwards, no research on agromyzids.
Okay, no, I am just asking about the conclusion. What conclusion you can draw?
Conclusion here, sir, very difficult to identify these agromyzids. We have identified up to 5 species on food legumes and up to species level based on the morphological characters. This is a conclude, sir.
What can be the extent of damage here?
No, we have not calculated extent of damage, sir. It is a taxonomic, purely taxonomic work.
Okay.
Because incidence we have not calculated. Here particularly we have collected based on the males and females we have collected and we have identified up to species level.
Okay, thank you.
Thank
So
you.
the next poster ID S4-31, a rust possible threat to chickpea cultivation in Western Maharashtra by SB Letake from Pulses Improvement of Project MPKV Rahori.
I have one question here. Hello, is there anybody
Yeah.
from the side?
Yeah, Letake or anybody from this poster? I think nobody, sir, is there.
Okay.
So the next poster is that S4-32 association mapping of fusarium wilt resistance in chickpea by Uday Chand Jha from IPR Kanpur.
Okay, somebody is here from Kanpur. Poster.
Dr. Jha or anybody from his team.
Okay, so nobody is there, then you can move.
Big team, but nobody is there.
Okay, we move the next poster. S4-33, evaluation of eco-friendly insecticides for the management of cram pod borer, Helicobacter parmigera in chickpea by N. Kamakshi from Regional Agriculture Research Station, Nadial.
I have one question for the.
Sir, please, sir.
Whether this finding is for one season or 2 season, madam?
Sir, this is for 2 seasons, sir.
1 year data or 2 year data? This is
2 year data, sir.
2 season, okay. 2 year data. So, what is your conclusion? Final conclusion? Out of what is another other biopesticides?
Actually, sir, in addition to biological control agents, one insecticide also evaluated, sir. Chlorantraniliprole is very effective, and in along with sir, dipel and one BT 127. It is a formulation from IIOR, sir. This is these two are on par with the chemical insecticides, sir.
How many sprays you have done?
2 sprays, sir.
2 sprays. Okay, thank you.
Okay, sir.
So the next poster ID is S4-36, emerging microbial biocontrol strategies against phytopathogens affecting pulses by Diksha Garg from Punjab Agricultural University, Ludhiana.
What is again difficult to read?
Sir, due to the paucity of time, I will move a little bit fast because the next session also we are having the poster session for abiotic stress management. So if judges allow, then I have to move a little bit fast.
Move.
So the next poster ID, I think S4-37, assessment of genetic variability for powdery mildew resistance in black gram by M. Tamizrasi. I think it is correct to pronounce Tamizrasi from Tamil Nadu Agriculture University, Coimbatore. So next poster S4-39, integrated disease management strategies against leaf spot and root rot disease of green gram by Manish Kumar from Agriculture Research Station, Mandur, Jodhpur. So next poster ID S4-42, integrating salicylic acid, vermicompost, and Pseudomonas species for management of chickpea wilt disease by Sangeeta Sahni from Department of Plant Pathology, TCA Dholi, Muzaffarpur.
Yes, Sangeeta is available or not?
Yeah. I'm Vishnu, I'm available.
Okay, how we can integrate all this?
Sir, what we have, first we have given the foliar spray of salicylic acid, and in the soil we have mixed the 15% of vermicompost, and the treatment was done with the PGPR. and we found that SAG has been activated. Here we have not shown the expression of PR gene, and we have also analyzed the 7 antioxidant enzymes, which shows that it decreases the chickpea wilt incidence.
Okay. Thank you.
So the next poster ID S4-44, identification of sources of resistance against Colletotrichum nindimuthianum, responsible for anthracnose disease in black gram, by Abhishek Gowda. It's from World Vegetable Center, South and Central Asia, ICRISAT campus, Hyderabad.
How many years they have studied?
Hello, sir. This is Abhishek Gowda. Actually, sir, this is the one year data, 2020, last year's, but we are now conducting same experiment this year also.
Okay.
This is the 1 year data, sir.
Okay.
So the next poster ID S4-45, refined genome assembly of Fusarium oxysporamodum, a causal agent of wilt disease in pigeon pea, by P.S. Shamang Deval and group from IPR Kanpur.
I have a question here. So for which race this genome was done, whole genome sequencing?
Good evening, ma'am. This is actually, we have not done the sequencing. The sequence is available in NCBI. The assembly submitted in NCBI is highly fragmented. And we have used different tools to assemble the genome in a highly assembled way to predict high number of genes.
No, my question is which variant or which strain has been used for whole genome sequencing, or it's only one isolate or many?
It's one isolate. It's a one isolate.
From Kanpur?
It's from Kanpur. It is submitted in MAU, NPBAM, MAU. So they have sequenced it. And we utilize the sequence to assemble the genome.
Okay, thank you.
The next poster S4-46, management of root knot nematode in pulses through biofumigation, by Dr. Bansa Singh from IIPR Kanpur. So next poster ID S4-48, chemical composition and bioactivity of spearmint volatile oil extract against Calasobruchus analis, by Sanjay Mbandi from IPR Kanpur. Next poster S4-49, assessment of brookid field infestation and then impact of field carryover infestation during storage in mung bean, by Ravanasiddha from IPR Kanpur. Next poster S4-50, a reaction of Ordbean germplasms to brookids, by Ravanasiddha from IPR Kanpur. -52, efficacy of different biopesticides against spotted pod borer, Maruca bitrata, in cowpea, by Kodandaram from IPR Regional Station, Dharwad. So the next poster S4-53, pod weevil, Apion emulum, Coleoptera apionidae, an emerging insect pest on black gram and green gram, by Kodandaram from IPR Regional Center, Dharwad. The next one is S4-54, response of mung bean genotypes for resistance against mung bean yellow mosaic virus, by Ravanappa from IPR Regional Center, US Campus, Dharwad.
Which strain is that yellow mosaic virus? Somebody is there?
Ravanappa. or Sable? I think nobody is there.
Go ahead.
Andaram can reply if he can.
So next poster S4-55, efficacy of biorational pesticide against aphid Apis crassivora, infesting chickpea by Ranjitha MR from CSAUT AUANT Kanpur. Next poster ID S4-56, eco-friendly management of dry root rot in black gram caused by Macrophamina fasciolina by Dr. K. Yamuna Rani from Agricultural College and Research Institute, Tamil Nadu.
Is there anybody to reply?
Yamurani? I think nobody is there.
Okay, go ahead.
So the next poster S4-58, identification of sources of resistance against dry root rot caused by Macrophamina fasciolina in mung bean mini core collection by Abhay K. Pandey from World Vegetable Center, South and Central Asia headquarter, ICRIS campus, Hyderabad.
How long you have worked? Somebody is there?
Yeah. Hello. This is Abhishek. Abhay is not here.
How many years you have worked?
This is, sir, 2 years data, sir. This one field condition is 2 years, 2018 and 19. And this paper towel method and sick pot method were conducted twice. And after confirming, we have went for the field conditions, sir, 2018 and 19 data.
Is there any correlation between the sick prot and your ragdoll method?
Yes, sir. Yes, sir. In table 1, we can see the sir. We have shortlisted 19 accessions from sick pot. Here we can see three accessions showing resistance in both field and sick pot conditions. Sir.
Okay.
So just following on question on that. So field condition, where had the study has been done?
Ma'am, it's in Esnin, Myanmar.
It has been done in Myanmar.
Yes,
So
yes.
it is under natural conditions or some?
Yes.
It's under the natural conditions for 2 seasons.
Ma'am, yes, ma'am, natural condition.
Okay.
Yes, ma'am. 2018 and 19.
So the next poster ID S4-59, integrated effect of Campoletis chloro-D and some natural farming inputs against Helicobacter parmigera by Pankaj Sood from CSK HPKV Krishi Vigyan Kendra, Sundarnagar, Mandi. The next one, S4-60, evaluation of integrated pest management module against pod borer complex in pigeon pea by Thilagam P. And I think affiliation is Tamil Nadu Agriculture University. It is written in the bottom.
What is again?
The next one is S4-64, transmission, nature and evaluation for resistance in black gram and trees against leaf crinkle disease by V.K. Satya from Tamil Nadu Agriculture University, Coimbatore.
Good afternoon, sir. Myself, Satya. Good afternoon, sir.
I have one question. Hello.
Sir. Sir. Sir.
What is the transmission method you have followed for studying this resistance? Which method you have followed?
Sir, the first level is the field screening, sir, under the natural conditions. So after that, resistant field entries were subjected to the artificial screening through sap transmission, sir.
Okay.
So through sap transmission. So in that VBG, sir, vector transmission, sir, sap transmission also. Itself, I got the infection, sir, so I didn't do the vector transmission. So this virus can transmitted through whitefly and also the aphids also, sir. Sir, for confirmation of the vector transmission, which vector is transferred the virus? So for that, I done the vector transmission. So whitefly and aphids both can be able to transmit the leaf crinkle disease, sir. And already that some reports are there, seed transmission. So that also I have confirmed. So for screening of the entries, I use the SAP transmission techniques. So in that 3 entries are found free from the leaf crinkle disease, sir. That is VBG 12053, KU 9158, MOSH 114. 3 are free from the leaf crinkle disease in the artificial screening also, sir.
So
Okay,
the
thank
next
you.
poster is S4-66, screening of different varieties of
sir.
French bean against Apis crassivora by Jacare from Department of Agriculture and Entomology, College of Agriculture, University of Agriculture and Horticulture Sciences, Shiva Monga. The next poster is S4-67, molecular characterization of virus species causing yellow mosaic disease of red gram in Andhra Pradesh by B.H. Chatyana from Regional Agricultural Research Station, Nadial.
How important is this yellow mosaic disease in red gram? So anybody is here from the group? Looks like no one.
I think nobody is there.
Yeah, you can go ahead.
So the next one, S4-70, efficacy of plant growth promoting rhizobacteria, simultaneous showed phosphate solubilization power and antifungal activity in chickpea by Vishun Dev Prasad from Bihar Agriculture University, Sabor. So the next one is S4-73, transcriptome analysis. Transcriptomic analysis revealed a complex regulatory network imparting yellow mosaic disease resistance in mung bean by Uttarayan Dasgupta from Indian Agriculture Research Institute, New Delhi. The next one, S4-74, occurrence and distribution of different diseases of lentil in lower Gangetic plains of West Bengal by Huma Nazneen from
Good evening, sir.
Bidhan Chandra Krishi Vishwavidyalaya, Mohali, West Bengal. So the next poster, S4-75, chickpea bacterial endophytes as antagonist against Fusarium oxysporum sciceris by Jackie Lanky from Punjab Agricultural University, Ludhiana. The next, S4-76, effect of meteorological parameters on population dynamics of pod borer in pigeon pea by Binay Kumar. I don't see the affiliation. The next, screening of pigeon pea genotypes against Striatum mosaic disease in Tamil Nadu by Kartiba from Department of Pulses, Center for Plant Breeding and Genetics, Tamil Nadu Agriculture University, Coimbatore. So the next is S4-82, morphometrics of IPR isolates of Pasteurella penetrans using scanning electron microscopy by Jagdishwaran from IPR Kanpur. So the next poster, S4-83, Impact of Weather on Population Buildup of Port Borer in Pigeon P by Shrikant from ANGRAU Tour.
Sir, good afternoon, sir. Myself, Dr. M. Srikant.
How do you see the pod borer population or helicobacter population increasing or decreasing from this study, from this relationship of the weather?
Sir, madam, from vegetative stage, we are recording the adult population by erecting pheromone traps, madam. From 45th to 49th standard week, between those periods, standard weeks, we have recorded highest population of Helicoverpa and also the Maruca larval population. Whereas the Helicoverpa, both the adult and larval population we have recorded. With regard to Maruca only, we have recorded larval population at weekly intervals. We have recorded highest population 45th to 49th standard meteorological week, which corresponds to peak flowering stage of the crop. That is the second fortnight of November to first fortnight of December, madam.
Just quickly, which is the major factor, climate variable, which you found highly correlated?
Major factor is maximum temperature and RH1 and RH2, madam. RH1 and RH2. Whereas RH2 is negatively correlated, whereas maximum temperature and RH1 are positively correlated along with sunshine hours and evaporation, madam.
Okay, yeah, thank you.
So the next poster, S4-86.
Thank you, madam.
CA Pro DH2 imparts resistance against Ascochyta rabi infection in chickpea through fine modulation of proline P5C cycle under drought stress by Mahesh Patil from National Institute of Plant Genome Research, New Delhi. That's the end of this poster session. Thank you, judges.
Okay, thank you.
Hello. Hello, sir. Hello. Sir, hello, excuse me, sir. My poster was supposed to present in this session, I guess, in biotic stress, but my number wasn't displayed and my turn was not there. This is poster number S277.
Which poster?
77.
77. Okay, we'll see it and then we will see whether that can be possible to display in the next this poster session.
Yeah, thank you.
My poster was also not there, sir.
Please tell me the number.
PS Singh and Saxena Ravi Prakash Nair, sir.
Sir, S396 is also not there. Hello? Hello, sir? Hello? 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 Sir, hello? Hello?
Hello, sir.
Hello. Hello, sir.
Hello.
Hello.
Yes, sir.
Hello.
Hello, sir. Hello, sir. Sir, S396 poster is not there. S396. I am giving in chart, sir.
S396 will be displayed tomorrow.
Hello.
Okay, sir. S396.
These are under session 4.
बस S3 अच्छा okay.
Tomorrow you display.
Okay.
Tomorrow your poster will be displayed.
There is information for the participants. I think there is a bit of confusion.
Ankit? This one is not working. No. No, embedded express, like fourth. Still, poster number will come there. So, good evening to everyone. I'm Dr. Senthil Kumar, and I'm going to convene this poster session under the theme abiotic stress management. And in this abiotic stress management, we received 86 abstracts, and we have received 21 posters. And first of all, I welcome the panel judges, Dr. Bindu Madhavan. He's a senior advisor and technical head, MCR Foundation from Hyderabad, and Dr. T.N. Tiwari, and Dr. K.R. Soren, and Dr. Ramlal Jat. They are from IIPR Kanpur. Okay, now the posters will be displayed for 10 to 15 seconds, and I will request the judges to ask their queries or questions. So the participants and the judges will be unmuted, and participants can answer the queries. Okay, now I am moving to the first poster. I request the judges to note down the poster number, which is available at the right top corner. And this is not the serial number, this is the poster number. So poster number 5.1, it's the first poster, and assessing the effect of cold stress on chickpea. Anju Rani et al. From Punjab University and CSK HP KV Palampur.
One question from my side, Dr. K.R. Soren. I'm speaking Dr. K.R. Soren.
Please.
Hello.
Please proceed. You can
ask.
Yes.
Means how you have imposed the cold stress, whether it is in the control condition or in the in the open conditions, environmental conditions. They want to ask the author how they have imposed the cold stress.
Yeah, participants, please answer the question. Yeah, anyone is available?
Hello.
Yeah, actually participants are not there, it seems. Any other questions
Okay.
from judges? Okay, I will move to another second point. Assessment of chickpea genotypes under different sowing environments using stress indices. Sunil and co-authors from CCS Haryana Agriculture University.
Hello. Again,
Yeah.
there is the same question because in this they have used the methodology, the RBT, and they have assessed only for the 1 year. And
Okay.
once we are going through these posters, whether they have used any genotype which is already been proven or which is available in the literatures in the control.
The participants
Hello?
concerned the poster can answer. I think in this case also participants are not available. Ankit, nobody is right. They can unmute, no? Participants can unmute themselves, no? Okay. Then I'm moving to the third poster, and the poster ID is S5.3. Assessment of mung bean genotypes for heat tolerance. It is from Shikha Chowdhury and from Punjab University.
Good evening, sir. Am I
Yeah.
audible?
Yeah, you are audible, and I'm
Okay,
waiting
sir.
for the questions from judges. Any key questions from judges? Thank you, participants. I'm moving to the next poster. Poster number S5.4, and effect of drought mitigation strategies on growth, yield, and economics of PGNP. Jadhav and Kute from Ahmednagar.
Hello, sir. Hello, sir. Good evening.
Yeah, you are audible. I'm waiting for the questions from judges.
Okay, sir.
Is there any key questions from judges?
So what is the conclusion in this particular poster?
Sir, conclusion is that application of Pusa hydrogel at the rate of 2.5 kg per hectare with mulching with organic residues at the rate of 5 ton per hectare recorded the best result for the getting higher yield of the pigeon pea.
So have you calculated any economic on this application?
Yes, sir. Economics also we have got the BC ratio of 2.2 in case of this treatment, sir.
Okay, thank you.
Okay, thank you, sir.
Any other questions from judges? Okay, next poster number is S5.8, screening lentil genotypes for heat tolerance by Anjali Bharadwaj from Punjab University, Chandigarh.
Sir, author is available?
Yes, sir. I'm available.
Yes. This experiment is done in the field condition or in the control condition?
Yes, sir. It is done in the field conditions.
Okay, so you have done the late sowing for the
Yes,
heat stress. So
Yes, sir,
whether you have find any biomass penalty?
What, sir? Pardon?
And what you are planting in the late conditions, obviously
Yes,
there may be the less biomass.
Yes,
Okay.
sir. Yes, sir. Yes,
So don't you
sir.
think
Here,
that don't you think that imposing the already the weak plant under the heat stress that
Yes, sir.
will mislead the finding?
sir, here in the figure 2 of results, we can clearly see the decrease in biomass in case of heat stress. and the decrease in biomass was more in case of heat sensitive genotypes as compared to the heat tolerant genotypes.
No, what I'm trying to ask you, that obviously once you will planted in the late conditions, obviously the biomass
Yes,
will
sir.
be the less. And again, when you are imposing the heat stress under,
Yes, sir.
already the stress plant, already
Yes, sir.
stress plant,
Yes, sir.
don't you think that it will mislead the finding?
Yes, sir. But sir, we are more concerned with the yield of the plant.
Okay.
Yes, sir.
No, I mean whether we have any solutions without losing any biomass on the plant, and we expose the plant under the heat stress conditions. That's I mean. Do you have
Okay,
any mechanism?
No, sir, we don't have any mechanism like this. So if we are imposing heat stress, then there will be the negative impacts of it.
Okay.
Dr. Soren, I'm moving to the next poster.
Okay. Thank
Poster
you.
number S5.9, screening of mung bean for salt tolerance through germination traits. Parameshwaran from Assam Agriculture University, Jharkhand.
So here author has not mentioned which of the salt he has used for imposing the salt, whether it is the single salt or the multiple salt.
Yes, sir. Good evening. I am myself Parameshwaran. Sir, we used a complex, I think, integration of 8 salts, sir, such as NaCl and then Na2SO4, KCl, K2SO4, magnesium chloride, MgCl2, magnesium sulfate, MgSO4, and then calcium chloride, Cl2, and then we adjusted the pH with the NaCl, sir.
Okay, so in one of the slide, if you go to this dry matter, so
Yes,
have you
sir.
find like if you see the trend, even in the 16 EC, you are getting more dry mass.
Yes, sir. Yes, sir.
Or it should be reverse.
We got a high dry weight in 16 EC, sir, because the salts will accumulate in the plant matter, sir, dry matter. So that's why it shows a high dry matter weight at 16 EC.
So that is even more than the control.
Yes, sir. Sir, in control we find less dry weight, sir.
Okay, less dry weight, but if you see the data in the 16 EC or 12 EC, the dry mass is increasing.
Yes, sir. It's gradually increasing, sir.
Okay.
Because a high salt concentration will import more accumulation of salts, sir, in the plant matter.
So don't you think that high salt accumulation will hamper in the growth in the development?
Sir, we just screened 8 days seedlings, sir. So it's just a basic work so we done.
Okay.
Yes.
Thank you.
Okay, just I'm moving to another poster. Poster number S5.10, standardization of priming technology in chickpea under sodic soil conditions by T.N. Tiwari and D.K. Agarwal. Any questions from judges?
No.
Okay, then I will move to another poster. Poster number S5.11, studies on heat sensitivity in chickpea by Poonam Devi from Punjab University, Chandigarh. I'm moving to another poster number S5.13, salinity tolerance of Chenopodium salt blader treated black gram seeds by Vinotini from TNAU Coimbatore. Any questions from judges?
It's okay.
Yeah, then poster number S5.14, evaluation of stress tolerance orphan legumes for use in dryland farming systems across sub-Saharan Africa and India by Mamta Nehra from Agriculture University, Jodhpur. Okay, poster number S5.15, 16, Trichoderma haussiana, potent bioprimary wilt disease in PGNP under salt affected areas by Sonika Ponte from IIPR Kanpur. Any questions from judges? I'm moving to another poster number S5.17. Impact of heat stress in lentil genotypes growing in red and lateritic zone of West Bengal by Hina Gain from Indian Institute of Technology, Kharagpur. Poster number S5.18, ideal root phenotypes for combating abiotic stresses in mung bean by Muralidhar Husky and co-authors from IRI.
Sir, I having one question to the Murli.
Yeah.
Yeah, whether this experiment, what they have done under the the semi-hydroponic conditions, whether they have kept any replications on that?
Dr. Muralidhar Husky is available. Or any participant concerned with this poster? Dr. Soren, participants are not available. I can move to next poster.
Okay.
Poster number S5.22, identification of photoinsensitive lines in wood bean and their characterization by Vijay Lakshmi from IIPR Kanpur and co-authors from IIPR Kanpur. Poster number S5.23, physiological evaluation of mung bean genotypes for drought and heat tolerance by Manu, IIPR Kanpur. Poster number S5.25, study of root traits under terminal drought stress in lentil. Swati Priya from Kurukshetra University, Haryana. Poster number S526, influence of stress, moisture, mitigation sprays on a resultant seed quality in chickpea. Dada Gaudar and co-authors from UAS Raichur. Poster number S530, screening and variability study for seedling salt tolerance in chickpea land races. Sneha Priya from ICAR, IARA and IIPR. Poster number S531, approaches to mitigate abiotic stresses in pulse crop of arid and semi-arid region by Sweta and co-authors from Haryana Agriculture University, Hisar. Any questions from judges? Poster number S532, heat stress tolerance in wood bean, identification of donors based on physiological traits and field evaluation from Devjyoti Sengupta, IIPR. Poster number S533, genome-wide profiling of microRNAs and their targets in male sterile and fertile lines of PGNP by high-throughput sequencing and degradome analysis by Abhishek Bora from IIPR. Any questions from judges? As the last session, last poster we have completed, and I thank the participants and I thank the judges, Dr. Bindu Madhavan and Dr. T.N. Tiwari, Dr. K.N. Soren and K.R. Soren and Dr. Ramlal Jot. I thank Chairman poster session as well as the organizing committee of this web conference for providing this opportunity to convene this poster session. Thank you very much all.
Thank you, Dr. Senthil, for nicely steering the poster session. With this, now we come to the conclusion of today's proceedings. There are a few announcements before we depart. Tomorrow, the session will start at 9:30 sharp on the same Zoom links which we have provided earlier. So kindly ensure to join by 9:15 in the morning. The second session will be starting at 1:30 in the afternoon, and it will continue till 3:30. Sorry, it will start at 1 o'clock and it will continue till 3:30. 9:30. And the poster sessions on crop intensification, diversification, and resource conservation, then post-harvest management and secondary agriculture will be organized between 3:30 to 4:30 p.m. The valedictory session will be organized between 4:30 and 5:30 p.m. And with that, we will be concluding the Pulse Webcon 2021. With this, I would like to thank all the moderators, lead lecturers, then invited the speakers of the invited lectures, panelists of different sessions, especially Director General Icarda, all the judges of different poster sessions, all the presenters, and the presenters of different posters, as well as all the colleagues here at IIPR who have been helping in all these sessions. And with this, we come to the end of today's proceedings. Thank you. Have a nice day. Thank you very much. Bye-bye.